development of the magnetic technology to develop algae, we have achieved high purity Astaxanthin, more than 90%, within a few minutes, actually. And this is a promising technology for the development of this product for cosmetic applications, actually rejuvenating creams and sprays. But also, we have achieved alive algae, microalgae cells, for a long time, which helps in several other biomedical applications, such as magnetically targeted drug delivery, for example. All these things we will discuss today. I don't want to be too long. I will pass fast the floor to Mr. Petros Kokkalis, member of the Euro Parliament, to open this workshop. Petro, the floor is yours. Thank you.
Thank you, Dr. Christoforou, and thank you, and good morning, everyone, and thank you for inviting me. I'm sorry I cannot stay for the whole workshop because we're currently in the process of negotiating of finalizing the negotiation on the new transitional regulation on the Common Agricultural Policy. So I'm obviously not a scientist, and I'm a politician. I am a member of the European Parliament, and I'm a member of the Agricultural Committee and of the Environment Committee. So I would just like to give a broad view introduction on the direction of the European Union in the next few years regarding both the research, but more importantly, the quest for sustainability and CAP reform. I am very interested in your project and the way that you have innovated in facilitating the production of algae. And this is very important because the European Union has decided and has stood by its decision in incorporating the new Next Generation EU and the Recovery and Resilience Package to follow a new growth strategy, which is encapsulated in the European Green Deal. The European Green Deal is the effort by the European Union to transform its economy in a sustainable and just and fair manner, including the recovery. I am in the middle of the negotiation of the transitional regulation, and we have a transition in the Common Agricultural Policy, and we have a transitional regulation because the famous CAP reform was never finalized, and now we have to incorporate in the new CAP. We have to reform the reform, and we have to incorporate not only the European Green Deal, but the farm to fork and biodiversity strategies. These are groundbreaking strategies which aim to do pretty much what you guys are doing, which is to produce more with less and do better. Use the resources more efficiently, use them faster, use them in a more healthy manner. As you know, the Green Deal is capped by the climate law, which is being discussed these days in the Parliament and will probably be voted into law in the fall. And this requires the European Union to cut its emissions by to come to climate neutrality by 2050, which means that by 2030 we will have to cut our emissions by at least 55%, probably by 65%. And this is important to you because this is, and to everybody, of course, because this is based on the science of the IPCC and what is needed to stop the global warming. But the Green Deal is not only about the emissions, it's also about providing a safer, healthier environment for everybody. It is about, well, first of all, cutting the emissions, and then, of course, establishing a more circular economy instead of a linear economy. It is about reforming all kinds of mobility. It is about reforming the financial system so as to promote sustainable investment. It's about investing in research and development in all those areas, of course. And it's also about reforming the food system. Now, the food system is very much a holy cow in the European Union, and during the pandemic, we were able to examine its inefficiencies and its efficiencies, its strong points and its weak points. The European Union, therefore, is moving very fast towards a ground reform of the food system, which will include a target for 50% less pesticides, a target for 25% less fertilizer, a target for 25% organic farming, and in general, there is a seismic shift from the Common Agricultural Policy being an instrument to ensure a fair income for the farmers, to ensure healthy and safe food for consumers. This is a momentous shift. It also includes the obvious that agriculture has a tremendous role to play in halting biodiversity loss. Right now, agriculture is both it's both high in emissions, but also it's a main driver of biodiversity loss. So in this process, where we will have to radically transform our economic, political, and social systems within the next 10 years, this is something that has never been done before on this scale. The European Union is the forerunner. The Green Deal is the most comprehensive public drive towards this science, towards the transition that science demands if we're going to transition to a safer world, safer and more just world. So we have a lot to do, and a lot of it is based on efforts of consortia like yours. Horizon 2020 has been instrumental in the past years in cementing those alliances and those partnerships. And from what, as a non-scientist, I can understand, and I cannot really understand all the magnetic and nanoparticle stuff, but I can understand that you can grow more biomass, more algae in a faster, safer, and less energy efficient manner. And this is precisely, in a nutshell, what we all need to do in all sectors of production and society. And this is not something we can do without the science and without being pioneers in science. So we are going to see within the next multi-annual financial framework an enhanced role for research and development. And also, I'm sure you're aware that we have a very meaningful shift in Horizon 2020, where we're going to be organized in terms of missions. The next Horizon is going to not be organized in main subject. It's going to have specific missions, and these missions are designed to help the transition towards sustainable and digital Europe. So I think that was my general introduction to point out both the Herculean task that we have ahead of us and our capacity as front runners, and not only our technical capacity, but also our moral and geopolitical objective for Europe to lead the world in this necessary transition. So thank you very much for giving me the opportunity to address you, and I wish you a very successful workshop.
Okay, thank you, Mr. Petros Kokkalis. Thank you for spending time. I know that you have some double agenda today because you have another meeting, another webinar going on in your other computer. And yes. Then thank you for spending the time. I hope that Evangelo and Nick do appreciate your participation to this webinar and your support in the bioeconomy, and more specifically to the microalgae world and the project VALUEMAG. I will take a few moments now to share my screen and to make some introduction before giving back the floor to Evangelo and Nick. Maybe Nick, before I do so, you want to thank as well Mr. Petros Kokalis before he's leaving?
Well, actually, we all thank Mr. Kokalis for that. Nick, if you like to give also a salute.
Thank you very much as well. Your microphone again. I think I have some problem with my voice. So anyway, thanks again, Mr. Kokalis. And the view of European Commission and is very valuable for us and the research community and give the way sometimes to move on. Thank you very much.
Thank you very much. Bye-bye.
Bye-bye. Thank you.
Bye-bye. Johan?
Yeah. I take the floor a few minutes for the introduction, general introduction of BIOKET Digital Edition before we start, let's say, with the webinar of VALUEMAG. Well, ladies and gentlemen, thank you for being here in front of us. It is with a very shared feeling that I'm introducing today's program, a feeling of happiness because after all these 2 years of work, finally here we are delivering the fruit of our work, or at least part of it. And we have prepared some jingle for you to start with BIOKET. Well, happiness as well, considering the number of participants to this webinar. Normally, and I hope that the number will continue to grow up. We are already more than 100 connected to this webinar. The webinar is live streamed as well on YouTube. Then maybe there are some of you that are on YouTube actually following the webinar. But we were more than 200 registered to this webinar. And it is the same for the whole 3 days of this webinar, for a total of more than 700 registered people over the 3 days. It is a real success, and we thank you for this. But also some bitterness and sadness, because the sanitary crisis we are experiencing now has upset our entire calendar. And even if we cannot do anything about it, we feel very, very sorry. We have tried to do our best to adapt, to show a maximum of agility, to satisfy our sponsors and you. We appreciate for the understanding of those who have been with us since the beginning. Finally, I want to pay tribute to the whole team of IAR who worked hard on BIOKET to make it happen. Ladies and gentlemen, again, good morning. My name is Johan de Koning from IAR, the French Bioeconomy Cluster, and welcome to BIOKET Digital Edition. Before starting with the program, few words for our sponsors. We thank them all. They are indeed making a considerable effort, and without them, we would most certainly not be here this morning. First of all, a few words for IAR, the French Bioeconomy Cluster, the organizer. And for those who don't know IAR, we are pleased to play a short movie to illustrate our activities. Sorry for that, I went too fast.
IAR is the French Bioeconomy Cluster. Let's find out the 7 services we provide you to support your innovations. Tomorrow's solutions are being developed today, from ideas to endorsement and funding of your projects. Our expertise and strategic support will be there to maximize your success. Do you want to diversify your activity to reach a new market? Through our research and consultancy services, we analyze, explain, and help de-risk your business and innovation environment, enabling you to make strategic decisions and identify early adopters and clients. Bioeconomy is a European priority. Having been in Brussels since 2008, we can make sure your voice is heard. We are your ideal partner to access European funding. Innovative startup? Our Investors Club is seeking projects like yours. Thanks to our help, reach out to venture funds and raise the necessary funds to boost your project. Speed up your growth and overcome technological hurdles, thanks to the innovation platforms of our ecosystem. Our Bioeconomy Leaders Network offers you unique business opportunities. Meet your future business partners and develop your expertise at the many events we organize throughout the year. Conferences, business conventions, tech days, so many opportunities to increase your visibility and promote your activities. Grow your business abroad and give your ambitions new horizons thanks to our international missions and delegations. Brazil, USA, Canada, India, Southeast Asia, we will enhance your tech and business partnerships. In a changing world, we will help you adapt your training courses to the needs of bioeconomy. Access tailor-made skills, from the endorsement of your training classes to posting jobs. IAR services are here for you. Find out more on en.iar-pole.com.
2 years ago, we were in Strasbourg, in the region Grand Est. This year, we were supposed to be in Lille. We thank our main support, the Regional Council of Hauts-de-France, and we hope to be there next year. We have as well some industrial sponsors, the platinum ones, Genopole and University of Lille. The Genopole in every leading biotechnology platform in France, quite discreet, but very effective. They are active in the medtech sector, but also industrial and plant biotechnology. There is where companies like Global Bioenergy, Insect, Abolis, or Innovafit, amongst others, are located. University of Lille, a major university in France with more than 72,000 students, ranking in top 30 of most innovative EU universities. And we like to thank in particular Franck Duménil from the UCCS, a leading authority in the world of catalysis, with in particular his high throughput screening platform, RealCat, a unique tool in the world of catalysis. We have as well some gold sponsors, amongst which we find the Tree Innovation Tour. The development agency of Dunkirk was proposing the visit of the port of Dunkirk with Dodry van Korenberg, a specialist on vegetable oils recycling, oleochemistry and biofuels, together with Biotifuel, the Total's pilot unit producing from lignocellulosic biomass via Fischer-Tropsch processed biodiesel. The second innovation tour was proposed by Lanolin Stella, a subsidiary of the group de Wavrin, which also includes Isis Pharma and Alpol. Lanolin Stella is one of the world leaders in production of lanolins. They are eager to show their new molecular distillation unit and installation at your service for tailor-made production. The last innovation tour was proposed by Pivot, a platform to industrialize bio-based processes, technologies, and products. They have everything to host the project of your choice and to industrialize them. The other gold sponsors are VALUEMAG, the BBI project that you will learn everything in a few moments during this webinar. Tournaire, Optimus, and Tami Silva, three engineering companies at your service for equipment, processes dedicated to green extraction and purification. P&O, a leading consulting company for your R&D projects. Accents, a French technology transfer company which markets many processes to transform biomass. They have an impressive portfolio of technologies. Then, last but not least, ARD, no need to present them. They are so much known in the field of process scaling up, in particular in industrial biotechnology, with their 220 cubic meter fermenter, a size which allows them to make industrial production to order. Their industrial culture and experience in both biotechnology, green chemistry, makes them the ideal partner to industrialize your process. We continue with our silver sponsors, Orodia Novacell, specialist in purification, L'Oréal, the new name of the industrial subsidiary of L'Oréal, then Vito, a Flemish research organization in the area of clean tech and sustainable development, known for the H2020 BioHorizon project and its LignoValue pilot unit. Finally, we like to thank our bronze sponsors. I won't mention them all because to fear of forgetting one, but their support is very much appreciated. And you can discover them on this slide. They are as well on our website. Now it is time to talk about the program of today, of our digital edition, sorry. We will be together for 3 days with 3 webinars on the program. In a few minutes, we start with VALUEMAG. Tomorrow, at the same time, we continue with the webinar of the Carnot Institute Tribeca, a webinar dedicated to phenolic acid. And then on Saturday, we finish in the afternoon from 2 p.m. to 5:30 p.m. with Ibisba, a webinar dedicated to biofoundries and bioprocesses. We will be immersed in the herd of synthetic biology with the best actors in Europe and in the world. This webinar will be completed with a B2B for networking and business opportunity. It represents a total of 15 hours of virtual B2B over the 3 days. The platform is still open. You can register or complete your meeting schedule. Just follow the procedure on the screen. We recommend you as well during this morning to keep an eye on your mailbox. You might receive some requests during the webinar, and then please reply to them to make sure you don't miss any opportunity. Now, finally, we are happy to announce a few coming interesting events for the bioeconomy. Next week, in the scope of our Intercluster 3BI, we do organize a free live webinar dedicated to post-COVID opportunities for the bioeconomy. Three invitees to present. We have one person from the Vereniging Netherlands petroleum industry. I'm sorry for the Dutch friends. My pronunciation is probably not so bad. I did 10 trials, but it's impossible for me to pronounce very well. Then one speaker from Neste and one speaker from SABIC. To register, you just have to pass via the IRS website. It's open for everyone. In September, BIOKET Digital is back with another Horizon 2020 project, the project Enabling BBP, a project encouraging the creation of efficient, structured biomass supply chain for the production of bio-based products. Then, not to forget, the dates of even this fall, 2020, they are all digital. I'm talking about the World Bioeconomy Forum on September 10 and EFIB on October 5-6. And then in 2021, please save the date of our event dedicated to bio-based solution. The Plant-Based Summit is back next June, and it will be in Reims, in the Congress Center of France. We have a brand new advisory board, and we are building an original and much more international program than usual. We invite you all to submit your abstract. The call for paper, it's open now. And finally, if you should just select one event next year, don't forget the most important one, and we hope you are not going to miss it. BIOKET on site will be in March 2021, in Lille, and I want to see you all next year. Well, now it's time to start VALUEMAG. We are very happy to welcome Nick and Evangelo on the floor. Good morning, gentlemen. Welcome to BIOKET Digital, and happy to finally being able to organize your webinar. We are ready now for 3 hours of immersion. I will stop sharing my screen. And Nick, Evangelo, the antenna is yours now for 3 hours of webinar.
Thank you, Johan. I think we can start with our first speaker, Dr. Angelo Ferraro, who has been responsible for many things on this project. Angelo, I think that the floor is yours, and you have 30 minutes to present the results of VALUEMAG.
Okay, thank you, Vangelis. I'll start sharing my screen. I hope everyone sees my presentation. So, good morning to everyone, to all the registered participants. I hope you can hear me and you can see the presentation. If something is wrong, please stop me. So I'll start telling you the origin of the acronym of VALUEMAG. It comes from the title of the project, that is valuable products from algae using new magnetic cultivation and extraction technique. So we used value and mag. And this is the word VALUEMAG. And today I'm going to give you an overview of the project with the main results that we achieved. So here there is the scope, duration, and budget. Here I highlighted that as it was said many times, we are going to provide a new solution for microalgae production and harvesting based on magnetic nanotechnologies. But also for the extraction also on the traditional way using supercritical CO2. And we developed also in the framework of the project what we call the selective magnetic separation method. I will give you some example on that. The project was scheduled for 36 months, but because of all the crisis that we had just in the very let's say last months that the project was supposed to finish, we got an extension until next month, till July 2020. The budget is that we received is around 4.8 million. So the objectives of the project are to set up a method to introduce super paramagnetic nanoparticles inside the microalgae cell bodies, because this, let's say, nanocomposite material in general, the cell don't accept them spontaneously. There are some species in the world that they produce nanoparticles, but they are not photosynthetic, so we cannot use them. Then there is the immobilization of these nanoparticles, of these magnetic cells with nanoparticles, because here comes the real innovation. So we will create a sort of biofilm covering this biofilm with very thin layer of water that allow us to save a lot of water. But for that, we had to develop a specific photobioreactor with a particular shape, is a conical shape. I will show you images and also a short video. And then the extraction with traditional and with the, let's say, new magnetic separated method. Let's say here I will show you the participants to the consortium. We are 11 members from non-European countries, and the coordinator is the University of Athens. And I think I can go to the next. Yes, here there is just the work plan, very, let's say, brief summary. So we have two main pillars in our project, research and innovation pillars in our project. One is the magnetic cultivation using magnetic nanoparticles. And the second one is the extraction and the, let's say, algae products that, again, we will use in some steps also the magnetic nanoparticles. Then we have exploitation, management, and also the operation and assessment with all the LCA and let's say analysis that tells us if our technology is viable and economically feasible or not. So here I'm starting showing you the results that we got. The first step, of course, was the production of magnetic materials, the production of these nanoparticles that must be done quick and also in a cheap and safe way. We developed in the frame of the project two methods that were not used extensively to synthesize nanocomposite. One is the microwave-assisted synthesis of nanoparticles, where, let's say, the reagents together with the catalyst are mixed and then passed through the microwave oven to provide the energy, and then they are dried. We can use also some kind of biocompatible materials in order to make these nanoparticles accepted by the cell, because they must be biocompatible. And then the second method that we developed in the frame of the project specifically for that is the flame spray pyrolysis synthesis that gives us the possibility to produce high quantity of nanoparticles in a fast way. And then after synthesizing these nanoparticles, they must be introduced inside the cells. And this introduction is done through electroporation. It is a technique used in biomedical research, but the main limitation of the system was the batch-based, let's say, system, because on the market there were no system that could allow us to produce liters of magnetic cells. So we had to design from the scratch a system that allow us to continuously pump cell through the cuvette, get electroporated, and then go back to the, let's say, collecting tank. Here is another view, and I have more slides with more details about this, because we had to do a very extensive study of the shape of the cuvette inside. Actually, is this small tube. The volume is 1.5 ml, and we can pump this 1.5 ml in 15 seconds. So we have a tool that can allow us to produce almost endless quantity of nanoparticles. Towards the end of the project, we also made some 3D printed disposable cuvettes in order to get the sterility of the samples, because we use different strains of cells, so we don't want contamination. So we just put the metallic part on this plastic part that have been done with the 3D printed, and we get our electroporation clean and smooth. And now here I'm showing you what we said at the beginning, one of the main achievements of the project, the fact that we can make live microalgae cell with magnets inside. So here we have a pellet of cell that is attracted from a permanent magnet. Here we have also, these are the studies we have done in lab to optimize the growth. A magnetic part here, and you can see that it's green, whereas here is without magnet and it's not attached. Here another view, another view of the vessel where we have the magnetic particles attached and they can grow. And this will be the principle that we will use on the cone, and is using on the cone actually, where we have a biofilm. Of course, we will not put all this amount of water, but a very thin layer of water. To prove that the magnetic particles are inside, we used, let's say, an indirect method that is the Prussian blue. So it's a reagent that in general, when there is iron around, it turns from yellow to blue. And actually, as we can see here and in many pictures, the fact that we have a blue inside the cell, this means that there are nanoparticles inside. Here another test that we have done to optimize the condition of transfection. We used some RNA oligo that are fluorescent. In this slide here, in this picture, we have stained the DNA of the cells with X that gives blue. The red is just autofluorescent, so it's background. And the green is our oligo that entered the cell. So we are confident that our cuvette works, that our protocol to make a magnetic cell is reliable and repeatable. Now, the cells, as I said at the beginning, do not accept the nanoparticles spontaneously. So we had to make a sort of pretreatment, and this pretreatment is the partial removal of the cell wall that is an hard shield that the microalgae cell have on the external surface. This is done by cellulose and other sugars, and actually we used some enzyme able to remove or partially remove the wall from the cells, and we made a lot of experiments doing treatment with the enzyme for 4 hours, 8 hours. Here I'm showing 4 hours and 16 hours. And as you can see, the cells are damaged because when we use then the protocol to make the sample visible on the SEM, there is vacuum, so the cell collapse because they are not anymore rigid. They lost their stiffness. We also used a mechanical treatment. We used glass beads in order to remove, but it was too aggressive and the cells were dying. So after this processing of digestion with the enzyme that removes the wall, the cell go through electroporation. And this makes a lot of stress to them. So we had to develop also a protocol to recover them. And actually what we have done is to enhance, let's say, add to the medium but this is just the magnetic cell, not the cultivation. So this glucose or fructose or other organic carbon that we put only in the recovery period, so for 1 liter of cell more or less. And we saw that after 4 days, they were able to grow properly. Here we have a decay of the growth because they were in batch. So they reached the maximum saturation point and they start to die. But we now know that after evaporation, we have to keep them in glucose, medium, 1%. And after 4 days, they can be seeded on the magnetic cone. Now, let's see the magnetic cone. The partner that built and is running the prototype for the cultivation is in Cyprus. So we have this warehouse in Cyprus where we have designed and built the cone. Here there is, let's say, just a simple drawing. We have the cone with the surrounding chamber. And the chamber is gas tight, so we can inject CO2, we can control temperature, humidity, there is illumination through LED lamps, oxygen also we can control, and all the environment can be sterilized with UV lamps. And of course, we have windows for visual inspection and a door in order to enter. And here, some screenshot of the construction of the cone that is 6 meters high and 6 meters in diameter. So we have a skeleton of steel, then some metal foils to hold the magnetic foil, and then an hydrophilic, let's say, foil where the cells are attached, physically attached. Here, just a more detailed image of what I just tell you. So we have a multi-layer, let's say, skin of the cone, where the cell finally will grow in this plastic surface with hydrophilic properties, because otherwise water was not going to spread uniformly. Many parts of the photobioreactor have been constructed or designed and then shipped in Cyprus. Here, for example, this is the watering system that was designed and built in Austria, and then it has been shipped in Cyprus. A lot of electric and hydraulic parts have been designed and constructed in Spain and then shipped in Cyprus. And here, just a little view of the technology that we use for the watering. We use a cross-flow technology that allow us to have let's say, a flow of filtration that never stacks. And the watering system uses filter with cross-flow filtration technology. Here is now the whole system that is assembled. You can see here, this is the chamber that actually is 7 meters high. We left a little bit of distance between the tip of the cone and the roof of the chamber. All the external pumps and tanks and controller are outside. So this is the inside view of the chamber. We have just lights. These are the LED lights, the cone, and the outlet tube and the inlet tube, and nothing else. Inside, of course, users or operators cannot stand, because here we will, let's say, there is an high concentration of CO2, because this is another innovation that we thought is the fact that we will not mix water with CO2. So we don't need to pressurize the system in order to provide the CO2 to the cells. But the cell will absorb, let's say, CO2 spontaneously, because all the air here is saturated with 1 or 2% of CO2. And we have electronic control. I will just make a magnification. So every pumps and valves can be automatically or manually controlled from outside, from the user. And of course, the system can work automatically 24/7. Now here I have a small video. There is no audio, or well, there is a audio, but it's just noise. I will try to start it, where you can see that water comes from the top. It forms a pretty good smooth flow. These are testing, let's say, attempt. And this is the flow that is covering the macroalgae cell. Here there is no cell inside because, of course, to take the video, we could not inject CO2. And this very thin layer allows us to save a lot of water. Because finally we can cover this cone with less than 100 liters of water. And theoretically, and also practically, because from the test that we have done until March, because then we had to shut down the reactor, we got a good production of cells. And this allows us to now set a new running test during the month of July. Another part of the project was the possibility to reuse the water and the CO2, because I repeat, in the chamber, we just put CO2, not pressurized CO2. So any kind of industrial process that produce CO2, after, let's say, cleaning, if there are other gases, of course, we cannot inject it, but we don't need pressurized CO2. It can be directly injected in the chamber without any other pressurized or depressurizing, let's say, step. So we analyzed the CO2 and the water reuse. And for that, this has been done in Italy from Second University of Naples. They have two small photobioreactors where they grew the cells with different concentration of, oh, sorry, touched the wrong key. Where two photobioreactors where they can control everything, and they have done growth trials with different concentration of CO2 up to 3%. And as you can see, the more CO2, the better the grow. But after the first grow, the cell were removed, and the same water was reused for the second attempt. Actually, what we have noticed is that the water cannot be, cannot perform as fresh water, because some elements, mainly nitrate and phosphate, are used from the cells, and this, even though we can inject CO2, impairs the growth, because we cannot reach, let's say, the same biomass concentration. Here we have around, at the plateau, around 2 grams per liter, whereas here we can reach easily 3-4 grams per liter. So CO2 can be reused. It can boost the growth, but water must be supplemented, let's say, after the filtration with some, at least nitrate and phosphate. And now we go through, let's say, the second part of the project, the second pillar of the project, that is the microalgae valorization, so extraction and use of this strain. In the frame of the project, we worked mainly on 6 species: Hematococcus, Dunaiella, Schendesmus, Nagropsis, Chlorella, Cryptoecodinium. Sorry for the pronunciation. I'm not sure that I have done it correctly, but you can read them. We worked mainly with the first four, and on them we have done a very deep analysis. These are just let's say indication. We have deliverables on that. Some are confidential, of course. We have to, if you need the information, we have to see one by one. But we have done a very good job on the characterization of this strength. And as we said at the beginning, both we managed to get a lot of astaxanthin, both with supercritical CO2. In fact, we can extract at 65 degrees and 550 bar nearly 95% of the astaxanthin, but also very good extraction from other valuable compounds like lutein, IPA, and PUFA that are fatty acids. Now I'll show you also another good result for which I'm personally proud. I didn't do this experiment by myself. They have been done from Enea in Italy, but we have used, let's say, extracts of Dunaiella to protect vegetables. Here, I'm going to show you what we have done. So we tested on tomato, zucchini, and tomato plants. These tomatoes have been deliberately, let's say, infected with a bacterium that in general they attack these vegetables. And then we put the solvent where we, let's say, dissolve the Dunaiella extract. And as you can see, the bacteria damage the fruit. But by spraying with Dunaiella salina extracts, you can see that the tomato recovered. So 95% of fruits are good, even though we infected them with a lot of bacteria. Here, the effect is much more evident. So in this case, with a scalpel, they have cut, let's say, in 3 points this zucchini, and again put some bacteria. But if together or later on we put Dunaiella salina extracts, the vegetable recovers, so the infection does not propagate a lot, and let's say shelf life of the fruit is enhanced. The same thing can be done on the let's say plant. So again, here the plant was contaminated with bacteria, and then we watered with Dunaiella salina extracts, and we sprayed with Dunaiella salina extracts. And actually, the treated is almost the same with the healthy control. Okay. And now let's go quickly towards the end of my presentation. And I'm talking to you about selective magnetic separation that also was a good piece of innovation, because it was used, but only for limited amount, in limited field of research, the use of nanoparticles to extract some kind of things, but we implemented it also for, let's say, valuable compounds. The idea is to take magnetic nanoparticles and to attach on it ligands, specific ligands that have high affinity for the target molecule. In our case, I will show you experiment for Astaxanthin. Actually, when we throw these nanoparticles in the tube with our target molecule, the ligand reacts with the target molecule. So the target molecule form a complex with the ligand and the nanoparticle, and they can be easily retrieved. So imagine that here you have a solvent. You just trap here the nanoparticle, and then you can easily pour away all the solvent. And there is a second step, an elution step, let's say, to remove then your ligand from the nanoparticle. And I repeat again, this is affinity ligand, so there is no covalent bond between the ligand and the target molecule. So in this case, we are sure that we can break this, let's say, quick bond formed between the ligand and the target with, for example, just a changing of pH. And here is the example of Astaxanthin. So here, 2 tubes where we dissolved Astaxanthin, and in 1 tube we just put the nanoparticles, let it react for a few minutes, and then put on the magnetic rack. So here there are some very strong magnets that can attract and can pellet the nanoparticles in the bottom of the tube. And as you can see here, we removed from the solution almost all Astaxanthin, 96, 97%, because then we used spectrophotometer to measure the ability. And of course, this is batch-based. We cannot think to make it in liters solvent. So we developed also a sort of magnetic separator. In this case, we have two prototypes. This is the simple one, let's say for small preparation, where the magnet actually is this tubular magnet that goes and a tube goes around it in order to enhance the surface, the trapping surface, where the nanoparticles with the target ligand remains attached, and then we can wash, or we can even just elute as it is. Here, the whole assembling that can be scaled up easily, because we can extend or put multiple, let's say, multiple magnets and multiple spiral tubes. So we can, let's say, isolate and purify a variable amount of nanoparticles. Then there is the second magnetic separator. This is my last slide. I cannot show you the system, real system, because are confidential information. But I can tell you that in this case we have two tanks. It can work in automatic. So there is a tank where there is the sample mixed with the nanoparticles, and then in this case we can even perform automatically washing and dilution, because with a series of valves, the trapped nanoparticles that are, let's say, with an electromagnet can be easily, let's say, manipulated. And in this case, we can get a high productivity and high purity, because we can, by setting the time of opening and closing the valve, we can even control the final concentration of our target molecule in the collector tank. So I finished. I will let you take note of the email address eventually you want to contact us, or the website where you can find more information, all the papers that we published, because up to now we have published more than 20 papers, scientific papers. There are more than 26 deliverable ready, and I think the work that we have done, it's great, and is going to be continued, because till the end of the project we will have another run of production of magnetic cells. So this is all for me. I will leave now the floor to Vangelis.
Yep.
I hope to get out. Yes. Okay.
Thank you, Angelo. Thank you for the nice presentation, showing all things, most of the things that we have achieved.
Yes, most of the things, because the limited amount of time does not allow to show everything, but exactly. More things will come later on, because we have also a video, for example, for the supercritical CO2 extraction and other things.
Yes. I think we can now pass the floor to the ENEA group, to Dr. Patrizia Casella and Dr. Antonio Molino. Ciao, guys.
Hello.
Ciao, Patrizia. I think you will give the speech, the presentation.
Yes.
Okay. Actually, the floor is yours.
Okay. Thank you.
And actually, we have some more minutes, actually, more than the scheduled time.
Okay.
Sorry, Vangelis, to interrupt you, because I just see that there is a question. It's only one. I think I can reply it.
Okay.
Is basically they are asking where the nanoparticles are located inside the cells, and if it affects the cell metabolisms. We know that the nanoparticles are inside the cells. They will form, but we don't know exactly where. Definitely, they form a sort of vacuole where the cell accumulates this nanoparticle. Because it's a foreign body, but it's surrounded with a lipid membrane, so it's biocompatible, and it looks like a subcellular let's say, organelle like chloroplast or mitochondria. So the cell, we guess, accumulates in a vacuole also because in order to get enough magnetic force, the nanoparticle cannot be dispersed inside the cells. They must be all together in order to give enough magnetic force to be trapped from the magnetic surface. And about the metabolism, it does not affect because we have now cells more than 50 or 60 days with nanoparticles inside. They are alive. There is no problem. We even try to freeze in -80 and freeze them, and they are still alive. But we guess that after a certain period of time, because the mobilized cells, they will go through, let's say, a very long exposure to the light. So they will accumulate some kind of mutations, and finally they will not split anymore. So the metabolism is not affected, but they cannot be used forever. I see more questions coming. Okay, but we may collect them at the end because I see that people now get interested.
Exactly. I think
yes,
we can answer the
yes.
questions at the end, the
yes.
session of the workshop. I think we can give the floor to Dr. Casella, to Patricia. Patricia, please.
Your microphone, Patricia.
Okay, I hope that you can hear me and you can see the presentation and follow the presentation. Good morning to everyone. I'm Patrizia Casella. I work in Enea and I work in VALUEMAG project activities. I'm working division biotechnologies and agroindustry and in laboratory bioproducts and bioprocesses. I'm working within VALUEMAG project, and I will start to present the circular economy applied to valuable products from algae. My colleague Antonio Molino, that is the responsible person in Enea for VALUEMAG project, will be available this afternoon during the B2B meetings of BIOKET Digital. As well known, the bioeconomy encompasses the production of renewable biological resource and the conversion of this resource and waste streams into valuable added products such as food, feed, bio-based products, and bioenergy. As possible to see in this figure, by applying the bioeconomy approach, allows to introduce healthy, safe and nutrition food, resource efficient and healthy animal feed, new food, supplements. Provide new chemicals, building blocks and polymers and other materials with new functionality and properties. Provide bioenergy and biofuels to replace fossil energy. Develop new, more and efficient and sustainable agricultural and marine practice. Improve bioprocessing and biorefinery concept and develop new process technologies such as industrial biotechnology. Deliver solutions for green and sustainable chemistry. The consequences of this approach and concept are to contribute to mitigated climate change through the substitution of petrochemicals by material with lower greenhouse gas emission and of fossil fuels by biofuel, providing the most important renewable carbon source, that is the biomass. Last but not least, it is to bring new business opportunities, investment and employment to rural, coastal and marine areas, fosters regional development, support small, medium enterprise. On the basis of this approach, the action plan of the European Union is possible to estimate that the bioeconomy has a potential of 2 trillion euros in annual turnover and over 18 million people employed, of which 76% of employment in agriculture and food and drink manufacture. In the VALUEMAG concept, we have applied the circular economy approach. Microalgae is cultivated by an innovative magnetic system that Dr. Angelo Ferraro showed in the previous presentation, on which carbon dioxide is captured from the solution group and represent the carbon quota from microalgae. After this stage, a green and safe extraction process by using supercritical fluid extraction is able to extract thermolabile substances such as bioactive compounds, producing extract solvent-free. In this direction, during the VALUEMAG project, carotenoids have been extracted from the microalgae Dunaliella salina, rich in beta-carotene, Haematococcus pluvialis, rich in astaxanthin, and omega-3 from Nanochloropsis gatitana, in particular APA, eicosapantenoic acid. The extract containing high-value products are appropriated for the application in food and feed and in cosmetic sector. In addition, exhausted biomass is valorized and carbon dioxide is recirculated within plant, within CO2 supercritical fluid extraction plant. The impact of output of VALUEMAG, such as extracted and bioactive compounds, are framed in the current situation about the use of Dunaliella salina, Haematococcus pluvialis, and Nanochloropsis gatitana extract. Dunaliella salina extract, which is rich of beta-carotene, is officially used as a food and feed additive with the identification number E160A. Also is currently used as a skin conditioning agent and protective agent in cosmetics. Haematococcus pluvialis extract has only been authorized as astaxanthin rich of oleoresin, and only authorized as a novel food. At the moment, is not authorized as a food and feed additive. Only synthetic form of astaxanthin has been authorized as a feed additive with the identification number E161J. Haematococcus pluvialis extract is authorized as ingredient in cosmetic as antioxidant agent. EPA, eicosapantenoic acid, from Nanochloropsis gatitana, has not been currently authorized as food and feed additive, but only oil rich of APA and DHA from the microalgae Eulkenia and Schizochytrium are authorized as a food additive. But Nanochloropsis gatitana extract is actually authorized as skin conditioning in cosmetics. The application of exhausted biomass is proposed for the potential use as feed material. The potentiality of the exhausted biomass is given by the content in proteins, carbohydrates, and dietary fiber, and also in carotenoid and fatty acid methyl esters. The use of extract and exhausted biomass allows the exploitation of all the biomass produced VALUEMAG project, within VALUEMAG project, in a perfect circular economy approach. The objective of the work package 3, animal food, nutraceutical and cosmetic from algae, have been reached with the collaboration between the coordination of the project, National Technical University of Athens, the Italian University of Campania Luigi Vanvitelli, FUSAV Institute, the companies Iparella, Exergy and Vertec. And then finally Enea, that is the work package leader with the employment of 44.75 months per month. So a complete characterization of microalgae composition have been done in task 3.1. Antipathogenic activity have been investigated in task 3.2. Supercritical fluid extraction by using CO2 have been investigated at bench scale in task 3.3, at pilot scale in task 3.4. Selective magnetic separation have been investigated in task 3.5 by National Technical University of Athens, and process modeling and design and risk analysis and safety control have been investigated in the following task. The activities of task 3.1 have been focused on a complete characterization of chemical composition of Haematococcus pluvialis red phase for the production of astaxanthin, Dunaliella salina for the production of beta-carotene, and Nanochloropsis gatitana for the production of fatty acid methyl ester. The complete characterization have been done after the identification of suitable method and official suitable method where possible to characterize the complete composition. Also, not only the characterization, but also thermal degradability of astaxanthin have been investigated. and also pretreatment on microalgae biomass and the solvent extraction optimization have been done. Sorry. Oh, sorry. Okay. Have been done. The characterization of microalgae have been done not only on Dunaliella salina, Haematococcus pluvialis, red face, and Nanochloropsis for the production of carotenoids and omega-3, but also on other microalgae. The results, the obtained results, show as proteins and total dietary fiber are the predominant compound in chemical composition. Carotenoids composition have been investigated, and beta-carotene is the predominant carotenoid in Dunaliella. Astaxanthin is the predominant carotenoid in Haematococcus, followed by lutein, and lutein is the unique and predominant carotenoid in Scenedesmus salmeriensis, which is chosen for the production of lutein. Fatty acid and lipids have been analyzed, and Nanochloropsis show the highest fatty acid content and also the highest APA content. In addition, other results have been achieved. A suitable and smart method have been identified to the quantification of astaxanthin by spectrophotometer and for identification of astaxanthin isomer by ultra high performance liquid chromatography. Thermal degradation have been investigated at a different temperature on astaxanthin from 40 degrees to 80 degrees. At the maximum temperature of 80 degrees, a thermal degradation was detected after 1 hour around 14% of the degree. And after 16 hours, a degree of 30% was detected on astaxanthin. This investigation has been done to understand the best operative condition for the extraction stage. In fact, on the basis of this, we have chosen operating at a temperature lower than 65 degrees for carbon dioxide supercritical fluid extraction. Furthermore, mechanical pretreatment of microalgae biomass by using bed milling mechanical pretreatment improved then 30% of extraction yield. About task 3.1, the antipathogenic activity have been investigated against fungi and bacteria. First, an in vitro screening have been done against fungi and bacteria by using all the microalgae extract obtained by using different solvent. At the end, in vivo tests have been done by using Dunaliella salina extract on tomato plants infected by Pseudomonas syringae and tomato and zucchini fruit infected by Pectobacterium carotovorum. Dunaliella salina's extract have been chosen because we show a great and interesting antibacterial activity during in vitro test against the bacteria pathogenic strain Bacillus subtilis, Pectobacterium carotovorum, Pseudomonas syringae. The results about task 3.2 and in vivo test on tomatoes fruit have been done infecting tomatoes fruit by using Pectobacterium carotovorum. This bacteria caused the typical small water-soaked lesion on the surface of tomato. And the tomatoes have been treated by 2 different concentrations of Dunaliella salina extract, 10 and 5 grams per liter. After 48 hours, no soaked water lesion have been investigated on the tomatoes fruit, but on tomatoes treated with the Dunaliella salina extract. But after 96 hours, a disease incidence was recorded, but lower than solvent and positive control. Solvent and positive control are tomatoes infected by Pectobacterium carotovorum, but not treated with Dunaliella salina extract, and or only treated by solvent. So a strong antibacterial activity of Dunaliella salina extract against Pectobacterium carotovorum was detected. The in vivo tests have been done also to zucchini fruit. Zucchini fruit were infected by using Pectobacterium carotovorum and treated by using Dunaliella salina extract at the two different concentration, 10 and 5. Also in this case, after 96 hours, a disease incidence on zucchini fruit was recorded less than positive and solvent control in zucchini fruit treated with Dunaliella salina extract. Showing this great and interesting antibacterial activity of Dunaliella salina extract, we decided to analyze the extract with the aim of understanding the molecules that cause the disease effect. The results show a relationship between the concentration of beta-carotene in the extraction zone and inhibition zone, and this effect was also assessed using the beta-carotene standard to confirm its effect against these pathogens. Within task 3.3, optimization tests have been done on at bench scale of supercritical fluid extraction. After the optimization of operational condition for the extraction of Astaxanthin from Hematococcus, beta-carotene from Dunaliella, and EPA from nanochloropsil gaditana, the optimized conditions have been validated at pilot scale. The extract and the exhausted biomass obtained after the supercritical fluid extraction at pilot scale have been characterized. Now I'm going to show a video about the carbon dioxide supercritical fluid extraction by using the pilot plant lure that is located in Casaccia Research Center near Rome. Hematococcus pluvialis dry powder have been used to load the plant, and at the end, a rich colored extract is obtained. So, as aforementioned, experimental tests at bench scale have allowed to understanding the best operative condition used at pilot scale. Besides of pressure and temperature, the main parameter obtained at bench scale and used at pilot scale are the residence time of carbon dioxide in the reaction vessel and the ratio between carbon dioxide and dry biomass used in all test period. The extraction process was performed at a pressure of 350 bar, temperature of 50 degrees for Hematococcus pluvialis and nanochloropsil gaditana, and 65 degrees for Dunaliella salina. The carbon dioxide flow rate was 0.47 gram per minute for Hematococcus pluvialis, 0.31 kilogram per minute for Dunaliella salina, and 0.35 kilogram per minute for nanochloropsil gaditana. The total extraction time results variable for each microalgae: 360 minutes for Hematococcus, 240 minutes for Dunaliella, and 135 minutes for nanochloropsil gaditana. On the right of this slide, you can see the results, the obtained results. So from Hematococcus pluvialis, we obtained an extract, an amount of extract that was 235.4 grams per kilogram dry basis. Astaxanthin was recovered around 49%. Lipid recovery was 77%, and protein recovery was around 50%. From Tunaliella salina, we obtained an extract equal to 104 grams per kilogram dry basis. The recovery of beta-carotene was 59%, the recovery of lipid was 25%, and protein recovery was 47%. From Nanochloropsis catitana, we obtained 191 grams per kilogram dry basis of extract, 14% of lipid recovery, and 17% of EPA recovery. It was observed that the exhausted biomass still has a promising content of bioactive compound to be applied and valued. The exhausted biomass of microalgae Hematococcus pluvialis still contain 17% of protein, around 80% of carbohydrates, and almost all the total dietary fiber. The same for Tunaliella salina. The exhausted biomass still contain a great amount of carbohydrates, 29%, a great amount of proteins, 60%, and also a complete amount, a total amount of ash. The exhausted biomass of Nanochloropsis catitana still contain 11% of lipids, 43% of proteins, and 28% of carbohydrates. So now, in conclusion, in the framework of VALUEMAG project, it has been possible to investigate the possibility to use microalgae biomass for several end use like cosmetics and nutraceuticals market and as biopesticide. Green technology as carbon dioxide supercritical fluid extraction allows to obtaining high recovery or purity for biomolecules like carotenoids and omega-3, depending on their value market. The evaluation of antimicrobial activity of Pseudomonas syringae, Pectobacterium carotororum, have shown for the first time the effect of Tunaliella salina extract against the pathogenic strain for both tomato and zucchini fruit plants. So the process schema of VALUEMAG fit well with the circular economy and the closing of the production cycle. Thank you very much.
Thank you very much, Patricia, for your excellent presentation. You have one question, actually, but I think it is better to leave all questions, even those answered up to now by typing. I feel it's better to leave them for the end in order to initiate a discussion between the participants and the speakers. So I see also much more comments, and I do think that we should leave them for the end after the presentation of all speakers. Questions are there and will be answered. So you will allow me to give now the floor to Dr. Panagiotis Zouboulakis from Theracell, who will present the business-wise development of opportunities of pharma and cosmetic products. Panagiotis, the floor is yours. Panagiotis, you should unmute your microphone, please. Your microphone is muted.
Okay, yeah.
Okay.
Last moment problems.
Sure. The floor is yours.
Thank you very much, Evangelos, and thank you all for attending this final workshop for VALUEMAG project. So I'm here to discuss with you some, let's say, business development opportunities that were shown from the results of the VALUEMAG project. I'm here as a TheraCell Advanced Biotechnology representative. We are working under the Business Development Unit. A few words about TheraCell. It is a regenerative biotechnology company. It is based in the United Kingdom with a subsidiary in Greece. And the company focuses mostly on healthcare technology, design and management in the field of regenerative biotechnology. And it was established lately in 2013, 7 years from now, with the primary aim to develop and provide advanced biotechnological applications and services, as well as products. Now, we have to notice that TheraCell is one of the few companies that in Greece that successfully audited and obtained a WHO GMP certification in the field of cell and tissue processing and regenerative medicine applications. And we have clean rooms that operate in accordance with ISO standards in EU GMP medicinal products. They are specifically designed for the advanced processing of human cells. A few words about regenerative medicines. We include cell and gene-based therapies, which are emerging segment of the overall biopharmaceutical industry. Cell therapy is the prevention or treatment of human diseases by the administration of cells. These cells have been selected, multiplied, and manipulated ex vivo, which means outside of the human body. Now, if the cells are derived from the treated patient himself, then these are called autologous, where if they are derived from a donor, these are called allogenic. As can be observed from the right-hand figure, autologous therapies are anticipated to emerge more rapidly than allogenic, both to become viable alternatives to conventional treatments such as drugs. The areas of authority of TheraCell include the laboratory services in cell and tissue processing, R&D of new product services leveraging on expertise in the fields of molecular and cellular biology, genetics, biochemistry, and regenerative medicine, commercialization of biotechnological products, and finally, design and implementation of clinical trials. Currently, TheraCell has two products that have been launched in the market. They belong to the orthopedics, mostly area, and is close to approval for a new dermatology plastic surgery product, while we are running R&D at the latest stage for nephrology and oncology products. Just a hint for the orthopedics, the two major and successful products is the CARTIL-S and novel cell-based approach for diseases like osteoarthritis. This can delay or even stop the progression of the disease. And CONSORTIUM, which is a product with a gel-type autologous stem cell implantation. It is composed of cultured autologous adipose-derived stem cells, combined with a special natural gel that serves as a scaffold for local cartilage regeneration in the joint. TheraCell is part of a group of companies, Qualia group of companies, and provides its know-how in the commercialization process of high added value products, with a history in cosmetics, nutraceuticals under Qualia Consumer Health, also generic drugs under Qualia Pharma, pharmacogenomics under the new entry of iDNA genomics, and of course, TheraCell that we have been talking about. TheraCell has access to a broad and comprehensive network of collaborators, mainly research institutes and universities, not only Greece, but also abroad. We have to notice that in the latest 3 years has received in funding from national and EU programs, the amount of almost 8 million euros. So coming to our project, coming to VALUEMAG, as already stated and presented, the project has brought a disruptive technology on cultivation of microalgae and biomass production and purification, as Angelo has successfully shown before. A biomass that has a wide range of applications, from biofuels and animal feed to high added value products. Microalgae are currently promoted as a new source of valuable nutrients for human and animal consumption. They contain a plethora of biologically active compounds such as vitamins, carotenoids, pigments, polysaccharides, and polyphenolic compounds. Among them, carotenoids are a category of bioactive compounds with considerable interest due to their industrial exploitation. Nevertheless, still are some limitations and challenges, especially for the use of microalgae, which continuously give space and rise to technology improvements. If we can name some of them, is the enhancement of microalgae production and growth rate, the dewatering algae culture for biomass production, the biomass pretreatment procedures, the purification and isolation of bioactive compounds, and optimization of manufacturing process in all stages. And as far as you have understood from the previous presentations, VALUEMAG gives, if not a complete, a partial solution to many of these limitations. So you have seen the technology, at least for the cultivation. You have seen the cone in a video that Angelo has presented. The VALUEMAG consortium has obtained a revolutionary solution for microalgae cultivation, harvesting and exploitation of biomass in order to manufacture and purify high added value biomolecules in, let's say, in an affordable and for sure environmental friendly way. So what kind of business direction can we have from this project? This stems from the project results either as products ready to sell, if we could say that, or even technology to sell, the VALUEMAG technology, but also they give rise to new products for further research and development. The main pillars, of course, are based in the cheaper biomass production, which can reach up to 10,000 times of increase of production, and cultivation of rare algae species that we are aware that they produce high added value chemicals. So these are the two main pillars that we can focus on. You heard about Astaxanthin, and of course, this is the primary, let's say, product that one could focus on. It is a xanthophyll, in words of chemistry, it belongs to the carotenoid superfamily, and it has a wide range of applications in food, pharmaceuticals, and cosmetic industries, since it has a very strong antioxidant profile, high nutritional value, and many, many other proven health benefits. Up to now, you also heard in the previous presentation, only the algal and yeast-made Astaxanthin are approved for human consumption. And we are expecting new regulations that will probably forbid the use of synthetic, even from animal feed. So you understand that there is a rapid demand growth for natural Astaxanthin and, of course, its extracts. So it is regulated as a super antioxidant with a considerable antioxidant activity. It has the capacity to either reduce or scavenge free radicals. So this is the main mechanism of antioxidants. It may reduce oxidative stress and facilitate the human body to maintain a healthy state. With significant productiveness and increasing demand, Astaxanthin is one of the high-value microalgal products. In comparison to other known antioxidants, natural Astaxanthin is by far more powerful. Astaxanthin is also well known to possess several other bioactivities, such as anti-inflammatory. And these bioactivities are capable to prevent diseases like macular degeneration, rheumatoid arthritis, cardiovascular diseases, neurodegenerative diseases like Parkinson, or let's say even chemoprotective character for cancer. As we may observe in this slide, the main microalgal products of Astaxanthin, which are currently approved in Europe and by the FDA in the US, are shown with their application sector, their characteristics, and in some cases their price. Most of them are color additives for fish and farm animal feed. Let's see how the market is for Astaxanthin. The truth is that data show that the Astaxanthin market is expected to triple from 2014 to 2023. And as you see in the latest bar of the graph, there is also an expectation to double the market in a few years. In respect to natural, this is both synthetic and natural. In respect to the natural Astaxanthin market, there is an increase which is explained from several safety issues and concerns. These are associated mainly with the petroleum-based Astaxanthin, the synthetic one, the latest data for the superior activity and synergistic effects of the natural extracts. This is always the case for a natural extract, the synergy, as well as due to the, of course, the consumer's acceptance. In respect to the geographical area and market, the APAC, the Asia Pacific region, has the largest regional market as expected. At least this is data from 2016, and this includes countries as China, India, Australia, and New Zealand. The market in Europe is a key market for Astaxanthin, and this is mainly due to health and animal feed sectors. So, at final, we know that the Astaxanthin market is oligopolistic and is dominated by a few key players. If we have to state a few trends for the market, the Astaxanthin is gaining importance due to the bioactive profile of the molecule, the increased potential for industrial application, the rise in health consciousness, especially in the developed countries, the increasing consumer requirements on personal care and cosmetic products, the popularity and acceptability in cosmetic manufacturers, the approval of Astaxanthin by US FDA and EFSA as generally recognized as safe to dietary supplements, and of course, the lack of raw material for natural Astaxanthin and inefficient advanced technologies up to now, at least up to VALUEMAG results, are limiting the market. Market may be segmenting according to the source, the product, application, and origin. Regarding the source, it can be either natural, as we said, or synthetic. We have to state that the synthetic Astaxanthin has 20 times lower antioxidant capacity than the natural counterpart. And of course, we keep in mind that up to date, the synthetic one has not been approved for human consumption. Now, regarding the product, the dried algae is expected to dominate the market, principally because it is used as animal feed in this form and due to no extra procedures and low cost of the production. Now, the form of soft gels is interesting because it's projected to witness a growth of more than 20% over the next period. We know that serious agents can be ascribed to this development because soft gel is an easy way of administration and easy availability in a variety of doses according to the needs. Liquid Astaxanthin in a soft gel is easily miscible and preferred for several uses. Now, regarding the application, it is segmented into dietary supplements, food and beverages, cosmetics, aquaculture, and animal feed. The major market, as I stated, for Astaxanthin is in aquaculture and animal feed industry as a coloral pigment. Now, the latest numerous verified health benefits of Astaxanthin, coupled with the increasing demand for new nutraceuticals, both of them contribute to an exponential growth of dietary supplement segment. Finally, in the food and beverage sector, Astaxanthin has been granted approval by several international food agencies, including the Canadian Food Inspection, the European Food Safety Agency, and this will support and promote the industry growth. The same will happen also with the cosmetic and personal care products, where Astaxanthin finds applications in numerous products and associated therapies. So what can be the opportunity here is to produce and distribute customized natural Astaxanthin extracts. You heard that the major source that we have also tried and tested under VALUEMAG is Haematococcus pluvialis, using the magnetic photobioreactor and, of course, the selective magnetic separation system. This will bring a definite competitive advantage with significant reduction of microalgal production cost. So higher production rate lowers the cost. The produced extract, of course, have to meet the standards and requirement for their use as bioactive compounds, like APIs in novel food and dietary supplements, as well as medical devices and cosmetics. So this brings me to the introduction of one project that has been implemented during the last years in TheraCell, under the collaboration, of course, with other research entities in Greece, and where we had the possibility to test some of the extracts and other compounds of microalgal origin to try to develop a new medical device. So this has to do with a multifunctional nanosystem incorporating natural therapeutic agents for the treatment of osteoarthritis. What is osteoarthritis? Just a few notes. It is a chronic degenerative musculoskeletal disease. It ranks among the 5 major causes of disability, and more than 250 million people currently suffer worldwide. We have to note that there is no cure, only disease-modifying treatments, which include physiotherapy and lifestyle change, non-steroidal anti-inflammatory agents, injectables of hyaluronic acid and cortisone, and finally, surgical treatment. Recently, and as I have already shown from the products of TheraCell, autologous intra-articular mesenchymal stem cells injection has been proposed as a therapeutic alternative for treating osteoarthritis. So what is the aim of a new product that has also to do with algal-based bioactive compounds? It is a novel injectable long-acting medical device for joint osteoarthritis, combining natural bioactive anti-inflammatory agents in biocompatible and biodegradable matrices. So what we did was trying to screen several natural compounds for different anti-inflammatory activities and synergistic effect for inflammation stemming from osteoarthritis condition. We found out that molecules, either polyphenols or oligosaccharides, that they stem from several strains, algal strains, may have a significant activity, at least in vitro, over mesenchymal stem cells. And we show that this can have a good effect on the cartilage, together with the anti-inflammatory properties. So these compounds may be incorporated into matrices like the polygalactic acid or the chitosan, and this can bring to a final modified nanocarrier nanosystem, which can be injected into the cartilage for as a new medical device. And what can be the competitive advantage of this kind of product? Local administration to the joint, so not a systematic administration, high load of bioactive compounds and controlled release, since we have the nanosystem, greater protection of the active ingredients, multipath and anti-inflammatory activity, immediate pain relief, we want, functional articular improvement, and increased cartilage quality. And we have to state that this product may be provided either alone or in combination with cell therapy. What about the market? The market is comparatively huge. We are talking about almost $12 billion by 2025. This is the projection. And a compound annual growth rate of around 7% from 2018 to 2023. And you can see some main points from the US. You see that the average cost per person per year for osteoarthritis is more than $2,000. And you also see that 40% of adults with osteoarthritis are inactive, and 80 billion is the cost from lost work from osteoarthritis. So, closing the presentation, I would like also to state 3 other R&D projects that they have been developed from the results that we obtained from VALUEMAG. These are all referring to the cosmetic industry. So the first one is the skin lightening products. In general, lightening product market is accounted for around 4 billion in 2017. And it is expected to grow with a growth rate of almost 8% during the forecast period until 2026. The second one is the anti-aging market, the anti-aging products. Sorry, I forgot, I skipped to say that for the skin lightening products, we have found some melanogenesis inhibitors, in some cases tyrosinase inhibitors, like the DPHC, These are also bioactive compounds stemming from algal species. The anti-aging products, the skin protection against oxidations, I just described the very famous Astaxanthin and Astaxanthin, both carotenoids. The anti-aging market is huge. It is estimated around $50 billion in 2018. And of course, the growth rate is around 6% between 2018 and 2023. A very interesting project is also the development of new sunscreen products. We have indicated and found also in the literature several phytoconstituents, algal constituents like phyto-N and phytofluent that can act as UVA and B filters. And this is also a very good market since sunscreens are being increasingly used in beauty products. And the market is accounted for let's say $8.5 almost billion in 2019. And this brings me to the end of my presentation, and thank you very much for your attendance.
Thank you, Panagiotis, for this nice, interesting presentation, particularly if we achieve the re-aging process that you mentioned in your presentation, then it's a really good market opens. At this moment, our good friend from P&O, Philoklis Pilidis, is to present the exploitation of the VALUEMAG results. Philoklis, are you there?
Hello, good morning from my side.
Good morning, very nice seeing you today. The floor is yours now. You can start if you.
Yes.
Sorry, Philoklis. Before you start, yes. I think Panagiotis Zubulak should stop his share screening.
Yes.
Because I still see his.
Yes, screen. Indeed. Panagiotis. Okay.
Okay.
Thank you. The floor
allow
is
me to, first of all, Mr. Christoforou, thank you very much for the introduction. Allow me to share my screen with you. Just let me know if everything is fine or.
Everything is fine. If you can.
Excellent.
Yeah, good.
Okay. Good morning, everyone. Thank you very much for your participation on this webinar. My name is Philoklis Pilidis, working as grants and innovation consultant within P&O Consultants, located in Brussels. Today, as a representative of my company and as a member of the VALUEMAG consortium, I would like to give you some information about the exploitation of the VALUEMAG results, as P&O participated on this consortium within the dissemination, communication, and exploitation activities. I think I missed once again. Can you see my screen? Is it okay? Yeah. A small introduction about the presentation and what you will see today. I will give you an idea about the strategy that we followed, the activities and the exploitation during the lifespan of the project, starting from stakeholder analysis in order to identify the entities and the key partners within the microalgae industry. Then I will jump to a market analysis. Panagiotis on the previous presentation already gave you some information about Astaxanthin, but I will explain a little bit more about carotenoids as well. That can be extracted from microalgae, and then a list of conferences, events, and workshops that the VALUEMAG partners have joined. They have been involved in order to communicate and disseminate the results of the project. Of course, the key exploitable results, the interesting part, let's say, as we are at the very end of the project, and a useful tool that will help us, of course, in order to have a follow-up of VALUEMAG, in order to have a technological development over the coming period. Starting from the strategy, starting from the activities, I will give you in chronological order the way that we approach the exploitation of VALUEMAG. Initially, at the very beginning of the project, together with, of course, all the consortium members, we perform a stakeholder analysis in order, as I said, to identify the most active entities, research institutes, academics and industries that are involved in the microalgae industry, of course, in order to have also an idea, an introduction about the industry. We performed a market analysis focusing on value-added components such as carotenoids, astaxanthin, beta-carotene or lutein that can be extracted from a microalgae. And of course, we also identified state-of-the-art processes and technologies regarding production and harvesting of microalgae in order to be able to compare them with the proposed VALUEMAG solution. As I said, a list of the conferences, the key exploitable results, of course, those that allow us to go over the next step, and the development of VALUEMAG into some potential funding resources for the near future. Starting from the stakeholder analysis, first of all, we had to identify what type of activities we should expect within the VALUEMAG of the microalgae sector. As you can see on the screen, we try to identify 5 different types of activities, starting from the inbound logistics, where we have the culture and the supply of microalgae, then the operations and the outbound logistics, where you can find, as highlighted within the light brown square, the majority of the activities of VALUEMAG, where we have the growing, the harvesting applications through the photobioreactor, the proposed photobioreactor technology. And as outbound logistics, we identify the extraction, the purification, and further processing of components from microalgae in order to apply them within cosmetics, nutraceuticals, and domains such as food and feed applications. And then at the very, very last end of a value chain, of a microalgae value chain, we have, of course, the marketing, the commercialization of the products, marketing and sales and services, such as the distribution of the final product, where VALUEMAG has not been involved significantly. So this first categorization allows us to move over the next step in order to identify the profile of the stakeholders and their activities within the microalgae sector. As you can see on the table, we separated them within 3 layers, involving some partners on the operation part in layer 1, those that are related to the production of algae or algae-based molecules, and of course the technology producers and designers. Then on the second layer, we have those involved at the end of the value chain for producing products such as energy from microalgae, biofuels, the food and pharma industries involved within the field, or some water recycling, water resource and water treatment entities. And then within the third layer, stated as RD, we have the research organization and the technological centers that are involved within this research, within this report. The last categorization and the results that you will see within this presentation has been done based on 3 different fields. We categorized entities and academic institutes within innovators, those that have participated within innovative EU-funded projects, the inventors, those that own patents within the microalgae industry, and some business drivers, the most important, the most active industrial stakeholders, those that shape the future of the microalgae field. In order to identify these stakeholders, in order to categorize them, we use tools such as CORDIS the Community Research and Development Information Center to identify EU-funded projects related to the microalgae industry, such as VALUEMAG. We used internal platforms within P&O, such as Matchpoint and WISP, in order to identify partners within our portfolio that have been involved in this domain, and of course PatentScope in order to identify who the patent owners are in this field. A comment on this before I forward below. As we are talking about P&O is a European entity, and Cordis mostly focuses on the EU-funded projects. Of course, the majority of the stakeholders that were identified are coming from the European continent. We are not against the others, but of course we mentioned some of outside Europe partners, but the majority of them, the vast majority of them that you will see in the presentation are located in Europe. And then as a final step, we have interviews with some of these stakeholders in order to discuss the technological aspects of VALUEMAG, what's the current status in the market regarding microalgae and algal-based products, and of course some fields of future research and investigation. As you can see on the slide, we identified several partners within the innovators, either research institutes or companies that have been participated in EU-funded projects, and of course, on the second category, we have some of the patent owners within this field. A comment regarding the years, you see patents related to the years 2013, 20, and 2016. We are talking about those where the maximum amount of patents related to microalgae industry have been found. And of course, as the stakeholder analysis has been performed at the beginning of the project in 2017, we are talking about up-to-date projects till that moment. Jumping on the second slide, we have a list of business drivers, companies within Europe, mainly in Europe, that are involved in the microalgae industry. As you can see, within several layers, either in the production of molecules, of algae-based molecules, or technologies related to microalgae, or end users that produce or they are involved in the recycling process of the value chain. Something that I would like to notice, and it is quite important for the next slide as well, the interviews that we have performed, is related to the type of the entities. If you check on the very last column, you will see that almost 90% of the entities that we identify and 90% of the business drivers are small or medium enterprises, startups or spin-off, companies within Europe. And this is quite important, and I will explain you why on the very next slide. As we identify a number of active stakeholders, we picked 5 of them from different layers, from the previous layers that I mentioned to you. And we discuss with them what are the technological aspects, the benefits or the drawbacks of VALUEMAG that they foresee for the future, what is the current status in the market, and of course, which type of investigation, which type of research we should focus in the near future. So regarding VALUEMAG, what they mentioned is that, of course, the objectives of the project to develop a quite selective magnetic separation method, a fast and cheap harvesting process that allows reusability. First of all, the use of minimum amount of water and the reusability of the water in the system, and then, of course, the significantly lower price of the produced microalgae. So, as Mr. Christophorou and Panagiotis mentioned before, if we are able to manage and to decrease the amount of the produced microalgae, first of all, we will be able to create new opportunities, and we will develop the position of microalgae as a resource, as an alternative resource against other crops that exist in the market right now. And of course, a drawback of the project, a disadvantage of the project, something that was mentioned, was the size of the photo bioreactor, which is quite high, and it should be considered for future applications if we want to introduce VALUEMAG technology in the market. As I said on the previous slide, you need to focus on the type of entities that we have within Europe. We have, of course, mostly small and medium enterprises, startups and spin-offs, which shows the maturity of the market in Europe compared with other leaders such as Asia and more specifically Japan, where large companies already produce and develop products from microalgae. So for now, the European and national funding are the main weapons, are the main resource of money for the microalgae within the continent. One of the interviews that we have with Bioalgostral, a company located in Réunion Island, if my French accent is quite good, is interested in order to test the VALUEMAG photobioreactor, quite important in terms of weather conditions, as we will see in a while. And regarding trends, what the interviews stated was that cosmetics biostimulants can be promising for as an intermediate term, pharmaceuticals as a long-term trend, and right now nutraceuticals are quite advanced but competitive segment in the market. And of course, other applications that they could focus within microalgae are either anti-fueling paints or bioremediation by using microalgae as an extract from industrial side streams. So some conclusions regarding the stakeholder analysis. We managed to identify 38 active partners within the microalgae. Out of them, almost 14, a little bit higher than for 1/3 of the stakeholders are R&D centers and universities. As I said, an indication about the maturity of the market in Europe. Most active European stakeholders are located on around the Mediterranean, in France, Spain, and Italy. Also quite important, as I said, regarding the weather conditions, due to the fact that the algae production is related, at least in open systems, is related to sunlight. And then in the rest of the world, we identified USA, Japan, and Israel as the most active countries. We mentioned that the European market is not mature compared to the Asian market, where we have large companies with already developed products. And the interviews we had focusing on the innovations of the VALUEMAG technology, the state-of-the-art market trends, and of course the fields for future research and investigation. Then going to the market analysis, you already saw some results presented for astaxanthin. We also performed our analysis on carotenoids, beta-carotene and lutein, more specifically. What we identified were 4 main axes for our research: the dynamics, what is the size, the current size, and the growth of these products, the segmentation regarding the application, the potential application of these products, a regional outlook where the main markets exist in every continent, and of course market players that have been involved in the production and the supply of the products. On the second part, we try to identify production and harvesting technologies. In terms of production, we focused on open or raceway ponds and closed or photobioreactor systems. And in terms of harvesting, we focused on state-of-the-art processes, flotation, sedimentation, and we try to compare it with the proposed VALUEMAG technologies. Over the next 3 slides, the structure and the content is quite similar. I provide a lot of numbers about the products. Sometimes numbers are interesting, sometimes are quite boring, I would say. So I will try to summarize these the next 3 slides. Of course, if you need more information, the presentation is available for everyone and I can provide it to you. In general, what we see what we foresee over the coming 5 to 7 years period is robust growth of carotenoids, of astaxanthin, of beta-carotene and lutein production, with an annual growth that varies from 4% to 30%, 13, excuse me, 13. That varies according to the different product and of course the different applications. Right now, the majority of the products are mostly used for animal feed, for animal nutrition, but of course there is a high interest around the world for human nutrition, for food, beverages, the nutraceuticals, cosmetics and pharmaceuticals from natural products. The market almost on 55 to 60% is dominated by chemically synthesized components, but lately there is a transition to natural molecules extracted from alternative sources. Europe and North America, as Panagiotis said, are the superiors in the market. But over the coming period, over the coming 7 years, Asia Pacific should be considered the third largest market, first of all, because of the size of the population, and more second for the interest into natural products and natural resources. I will skip these 3 slides. As I said, it is full of numbers available for everyone if needed. And I will go to production technologies, the market analysis we performed for those. As I said, we focused on open systems and closed photobioreactor systems. Based on our findings, based on what you see on the table, of course, we can notice, we can mention the superiority, the dominance of closed systems over the open ones. First of all, because they are not dependent, excuse me, on weather conditions, which allows a better reproducibility, the better control and standardization of the process. As we are talking about a closed system, it is better to minimize any type of water or CO2 losses and, of course, the contamination risk. Conditions, reasons that increase significantly the period of, that minimizes, excuse me, the period of production, which for closed systems is around 2 to 4 weeks, and at the same time increases significantly 10 to 40 times the biomass concentration, which is approximately 2 to 8 grams per liter. A small comparison of open systems and photobioreactors with VALUEMAG. Of course, as we are still running the project, we do need numbers from our partners regarding the investment cost or the operating cost in order to confirm that VALUEMAG has benefits, offer advantages over the existing state-of-the-art processes. The same applies also for harvesting technologies. We identify several of them that they have significant advantages in terms of cost, in terms of, and disadvantages in terms of cost, the type of algae that we are able to grow and to harvest. I will focus more specifically on centrifugation and filtration due to the higher amount of dry solid material that we can harvest. And in terms of VALUEMAG, I will mention the minimum amount of water used on the installation that results to fast and inexpensive harvesting, and at the same time allows reusability of the water in the system. So once again, the numbers, I already gave you these conclusions in advance. As I said, a robust growth in the market for carotenoids and components, value-added components from microalgae, from 4 to 30% over the coming period. We have applications in the human nutrition, high interest in human nutrition, cosmetics and pharmaceuticals, the transition to natural components from alternative resources, and in terms of production and harvesting, the superiority of photobioreactors and centrifugation or filtration harvesting due to higher amounts of dry solid material. A list over here I will show you, and on the next slide I will show you a list of conferences, events and workshops that we as partners in total, not only P&O, have participated during the 3 years of the project. I apologize in advance if I missed any of the partners. It didn't happen on purpose. Of course, please let me know when this table will be updated accordingly. And then reaching closer to the end of the presentation, I want to present a table of the key exploitable results that we managed to obtain during these 3.5 years of the project. We identified and we separated the partners of the consortium into large companies involving P&O, SMEs and academics, and of course 5 types of key exploitable results: the products, processes, consultancy, knowledge, and network. I will talk in general. I will not focus on a specific partner. What I will say is that, of course, we were able during these 3 years to gain knowledge, to extract knowledge for, of course, important and significant products such as cosmetics, foods, nutraceuticals, knowledge about the chemistry and the technology behind the production and harvesting of microalgae, knowledge in exploitation, dissemination activities, or in general on the sector. Then this table, the key exploitable results, will give us an opportunity in order to discuss with the partners, in order to confirm with the coordinator, Mr. Christophorou, that what VALUEMAG is an interesting project, and of course we want to go further with it by using the results, by using the knowledge that we have for other project ideas. So at the end of the project, we have the VALUEMAG on technology readiness level 5, and what we are expecting, what we would like to see in the near future is VALUEMAG on a higher tier level, either through the BBI funding, as it was before, or the coming Horizon Europe call topics. Of course, 2 different approaches, either by transferring VALUEMAG on its own into a new project, or by transferring the knowledge, isolating the key exploitable results and the technology that we have into projects that these results will provide value, they will add value into the project ideas. Some interesting sources are, for example, the key accelerator phase 2, excuse me, the BBI Demo 3 project, quite convenient if we want to transfer VALUEMAG as such. The live proposals, especially on the environment and resource efficiency, and the New Europe Green Deal, focusing on area 6, from farm to fork, if we are targeting into food applications. Excuse me. Something that I would like to mention, and it is not stated on the slide, is right now, of course, we had our discussions with the partners, we had our discussions with the coordinator, and we are in discussions with other BBI projects, with other project partners, in order to bring synergies, in order to develop synergies, and to transfer VALUEMAG into a new challenge, into a new step over the coming months, over the coming period. A very last slide, I would like to thank all of my colleagues that have been involved within the project. Their contribution was significant, namely Same, Habib and Natalie for their work, and of course that those who have been previously involved within P&O and within VALUEMAG, that they are not right now in P&O. And I would like to thank you for your attention, and to apologize in advance for the lot of information on the slides. Thank you.
Thank you, Philoklis, for the very interesting presentation. Of course, we have to proceed for the exploitation of the project, for the continuation of this project, in order to reach the commercializing TRL to realize the products actually out of the microalgae cultivation process, and we are looking forward to that. Now I would like to ask to invite Mr. Luca Petruccelli from Vertech to present the life cycle analysis assessment of the VALUEMAG technology. Actually, Luca has a very nice view in the monitor with the VALUEMAG, with the big VALUEMAG.
Yeah, I can. Here it is.
Photo, actually.
Background, yes.
Yeah.
So,
background, yes. Sorry, this is it. So the floor is yours.
Thank you, and thanks for introducing me. I'm representing here Vertech Group, which is a small consultancy company based in Nice, France, and we are working all around sustainability assessment, life cycle assessment, and so on. So I'm taking the floor in place of my colleague Mathilde Fiorletta, that was the project manager of VALUEMAG project, and she actually worked on the LCA. I helped her supervising the work, but she did most of the work, and she prepared also these slides. So I'm going to share the screen and give you an introduction of what LCA is. I suppose that not all the people that are attending this webinar may be familiar with the methodology and the main results of the study. So let's see. I suppose you see now it's a full screen. What's happening? Okay. So
yes, it is full.
Okay, thanks. So as I said before, the main person to ask questions, if you are interested in this study, is Mathilde Fiorletta, and at the end of the presentation, there's also her contacts reported. So just a brief introduction. The LCA is a methodology that is meant to assess the impact of an activity throughout the life cycle of all the process that brings the final product or service in place, and is standardized by the ISO 40040 and 44. So it's part of the environmental management area in the ISO. So there are four main steps that are the goal and scope definition, the inventory analysis, so the input and output flows are analyzed to get the LCA model. And out of this analysis, there's a final life cycle impact assessment that translate the emissions and the use of resources into environmental impacts or indicator of potential environmental impacts. Throughout the process, there's interpretation and there's a reiteration of these phases as far as possible to improve the outcome of the assessment. So the main concept is that we normally, when we perform an LCA, the methodology says that we have to analyze all the input and output flows of an activity. And normally we translate these emissions, for instance, into impact indicator like the global warming potential that is measured in CO2 equivalent, thanks to conversion factors that are published by different let me say, institutions. And for the global warming potential, this is the IPCC, so the International Panel for Climate Change. But obviously, only the emissions at the production plant is not enough, or the activity where these take place is not enough to perform a life cycle assessment. We have to go back and understand what happens in the supply chain, what happens before, and sometimes, depending on the boundaries of the study, after the product is in use, during the product is in use, and after, to understand what are the emissions or the impacts that are happening beforehand and after the main production activity. So what happened is that normally what we call primary data is what we collect at the production site, and what we call secondary data is data coming from database. The one that is most known and used is EcoInvent, and this is help us in describing the supply chain for all the ancillary materials and energies that are needed for our production activity. One of the main things of life cycle assessment, and which is the value of these methodologies, that normally we do not compare activity with other activities or product with products. We compare the function of these products or services. So in the case of light bulbs, we do not compare just one light bulb with another. We compare the service that is provided by the bulb, which can be translated into a number of hours of light for a specific amount of luminosity or lumen. In this other example, this can be quite common for anyone, is the groceries bags that have to last for a certain amount of time and has to be able to carry a certain amount of weight. And so only by comparing these by functionality, we can understand which is the best options that are available to the consumer or to the user of a specific product or technology. So let's get into the analysis for VALUEMAG. And so we will follow the steps that I mentioned before and describing how the assessment of this process has been carried out by my colleague, again, Matilde Fiorletta. Scope and goals. So what was the main topic and the main scope of the assessment? First of all was to assess the sustainability of the process that the project VALUEMAG put in place. And we defined two main steps that we want to analyze and then compare with other existing technology. So the first is the cultivation of the algae, and so we had we have the model that has been elaborated from the data coming from the pilot site, and with the cultivation of 3 different types of algae. I beg your pardon because I'm not an expert in this topic, and so sometimes maybe the pronunciation of the way I mention the algae or other specific topics are not 100% correct. So the first was the cultivation of the biomass, and the second comparative assessment was the comparing these biomass that had been cultivated and the extraction of valuable substances out of this biomass. And so these are the two main steps, and this has been then translated into different functional units. As you can see in the foreground, the background system, different steps were included or not according to the availability of the information and availability of data in the databases. Which are the benchmark scenarios? So in the previous presentation, you've seen that there was a very thorough analysis of the market. We focused on what was available from the literature in terms of data to compare the VALUEMAG solution with the open ponds technology and tubular photobioreactors. These are two again, technologies, all these areas still an area that is under development. And so the data came from research activities and papers that were published on the efficiency and the LCA of those two systems. And the references are reported in the slides. So as I told you before, the two scopes were then translated in functional units. So which is the function of the system that VALUEMAG is putting in place? In this case is the cultivation and production of an amount of microalgae as biomass. And here we have defined as 1 kilo with a moisture content of about 85–95%. The second functional unit takes into account the extraction of the substances that are under study. So I will not pronounce them correctly, but I suppose it's Astaxanthin, beta-carotene, and EPA. So you see here how these substances are taken out from the algae biomass and which are the main steps for doing so. The system boundaries. So as we said before, system boundaries are critical in the definition of the LCA to understand what we are comparing, and the boundaries were set as cradle to gate. So what we compare is what is needed to produce the biomass, and then what is needed to extract the substances out of the biomass, but then we stop there the analysis and we don't do any other assessment further than that. So we stop at the gate, so at the end of the production system. The geographical scope is where the biomass have been grown for the pilot site, so in Cyprus, and the supply chain that we took into account were on average European production. So all the raw materials were considered as coming from an average European supply, while the electricity system is dedicated of the Cyprus. As you can see, we have all these materials that are coming, so the nutrients, electricity, and water supply into the system. The system is split in 4 main subsystems for the assessment. So as you may understand, the complexity of this research for the LCA purposes is hugely simplified to then analyze this single subsystems as a black box where we have input and output. For this, the main outputs are obviously the emissions to the environment, as well as the products as biomass and olefin. The bioproducts that are produced by the system as animal feed, and then the overall waste and wastewater that goes to treatment. So getting a bit into the different systems, we can look at the inventory. And so the first is the magma production, so the magnetic microalgae production, and the introduction of the span into the algae. This is a process that is then periodically performed every 90 days. And then the magma solution is applied to the SOMAC, so the conic system for growing the algae. So here you can see the mass flow and the energy flow that has been analyzed for all these systems. The second step is the cultivation of the microalgae. So here we have the magma coming from the previous system. And then we have a mix of nutrients and oxygen and CO2 that enters into the system, as well as water that is then recirculating with the dewatering system, and almost half of the water is then recirculating into the algae cultivation. We took into account that the different strain of algae that has been cultivated requires a different mix of nutrients. So what you see here is the different mix of nutrients and output that changes according to the algae that is under investigation. And then part of the microalgae that is cultivated goes back to the magma production. The third step is the pre-processing. So here the algae has been cultivated and then is pre-processed to be then prepared for the extraction. There's the watering, and then there's energy required to perform the spray drain and the steam explosion. The quantity of the dried biomass is depending on the type of algae, as well as on the quantity of algae that has been produced in the previous phases. And at the end, we have the extraction of the substances. And here, this is the innovative process of the process with supercritical fluid extraction. So here we have the different types of algae entering the system and the quantity of substances that can be extracted from the different strain. The CO2 is recycled, but there are some losses in the process, as you can see from the balance of the flows. Okay, so this is the in very short and synthesized way of describing the technology from an LCA point of view, and all these inputs and output have been then calculated as potential impacts. So here what we see is the classical impact indicator of potential impacts that are normally used in the LCA, that are climate change, photochemical ozone, acidification, eutrophication, etc. And in the case of algae, one of the critical aspects are obviously the land use and the water scarcity indicators that are very relevant for this typology of production. In green, you will see the 3 different microalgae that are the scope of the project, and the 2 microalgae cultivation systems that we mentioned before, where we have the open pond in yellow and the tubular growing in blue. So you can see that as so far, the pilot plant compared to these other 2 pilot plant scale production is very promising for all the indicators, except for the acidification, where we have a slightly higher impact compared to the open ponds. That is mainly due to the specific electricity mix that we took into account. Sensitivity analysis on this is also reported at the end of the slide. There's a lot of noise in the background, so I don't know if there's anyone. Okay, thanks. Here we have the details of what are the main contributors of each single indicator for the different systems. So the first 3 are again VALUEMAG, and you can see that it's always showing a lower impact. And for climate change and acidification, the gray area is mainly coming from the electricity. So the electricity shows a lower impact because there's generally a lower requirement of electricity, but the specific electricity mix of Cyprus shows, compared to others, a bit higher contribution to the acidification. This is mainly due to the fact that the Cyprus energy mix is based on fossil fuels. And the other interesting part that we can show is that the other 2 systems, generally they require higher energy consumption, and this is visible again in climate change for the open pond. This is because of heating process. So the open ponds are open and they were based in Germany. So to grow the algae, the heating was needed, and then was showing a relatively higher impact compared to the other solution, and especially with VALUEMAG. And while the specific nutrients that we use for the tubular growing system is generally showing a higher impact to the other indicators and to the other systems. Okay, so first good outcome of VALUEMAG that has been analyzed with the LCA is that the VALUEMAG solution shows real benefit compared to other existing technology. Then the second step is the production of the substances that are extracted from algae. And so in this graph, you can see which is the contribution of the extraction system compared to the previous steps for all the indicators, where the previous step of cultivation of the algae is reported in gray. So in gray, you have all the steps from 1 to 3. And then the 4th step is the extraction itself, which has different contribution that are the treatment, so the carbon dioxide liquid, the heat needed, and direct emission of CO2, that is the escaping emission of CO2 that is impacting the climate change for the CO2 extraction. In this case, we can see that the process is then extracting the different substances like the EPA, the astaxanthin, and the beta-carotene, and this is reported the difference between the impact that is changing according to the efficiency of extracting this substance from the algae, since if I'm not wrong, the process of extraction should be fairly similar for the tree. And here we report the difference in between the three substances with climate change, but the other indicator shows a very similar trend. So the last part is comparing the impacts that are the three substances are showing compared to other routes for producing the same substances, that they're still in progress, and we are hoping to finish as soon as possible this analysis, because we are still need to make sure that we are comparing the same quality of the substances, etc. But what we know for sure is that at the moment, beta-carotene compared to the extraction of beta-carotene from carrots is showing very promising results. The production of astaxanthin, then this is again is also compared with some available information, and again is showing very good results, at least more than 10% in reduction of impacts for global warming, eutrophication, and even more for these other two indicators that are acidification and photochemical oxidation. So the EPA is not yet analyzed. The final step is the interpretation. So generally, we've seen that obviously electricity and the energy consumption is one of the main contribution for all categories, and has been said at the very beginning of this webinar. The aim of VALUEMAG was to create an energy efficient system for producing bioalgae. The magma production seems to be a negligible contributor to the overall impact of all processes. So the extra materials that are needed and processes that are needed to allow this innovation seems to be negligible. The growth rate of the algae is a key factor. So at the moment, as we said, this is a pilot scale, and probably the upscaling will be even more promising, but further analysis are needed. The sludge, so the biomass production seems to be very good, and this is happening in Cyprus. We don't know if transferring this technology in other countries at the moment. We don't know. I would say at the moment, the analysis that we're carrying out, we are not aware of, but probably other partners may be saying something on this. What may be the differences of transferring this technology in the northern area of Europe, and if probably more energy may be needed for heating. This we didn't do any assessment on that, and this for the LCA. Beta-carotene and from carrots and astaxanthin production has shown anyway a very good outcome and reduced impact compared to existing technology. And further investigation for beta-carotene extraction from algae and EPA, again from algae, needs to be investigated a bit more. Just the last slide. This is sensitivity analysis on the electricity mix that we investigated, assuming that there are no changes in the energy consumption for the different location. And so as you can see, the zero represent the current situation of the pilot plant in Cyprus, and we played around with the changing the energy mix of different countries to see how the impact will change accordingly. And you can see that generally other countries that have more favorable energy mix because of more let me say, renewable energy used for energy production is available in the country, but other impacts may increase as well. And this is because, again, from the mix, like in Germany, where we have a lot of coal, so eutrophication of fresh water may be worse. So this is a fairly overview of the work that Mathilde put together and followed the project in the last couple of years. So I'm very happy to had the chance to present. She had a clash in the calendar. If not, she would have presented herself. She would have been very happy to do so. If you have any questions on this specific study, please do not hesitate to contact her. And this is me. I will be happy to answer any questions if I can at the end of this session. So I'm stopped sharing and leaving the floor to you.
Yeah, thank you very much, Luca, for your nice presentation and the nice results concerning life cycle analysis for the VALUEMAG project. With this presentation, the presentations related to the VALUEMAG project have finished. Now, we have had two invited speakers to present related work. One is Dr. Natasha Bosic from the University of Belgrade as an invited speaker concerning a study on the comparison between the bioethanol generation and hydrolytic enzymes engineering. And the other one is Dr. Yasmina Vidic from INRA, France. Unfortunately, Jasmina will not be with us today. She couldn't make it. She would speak about bacteria and related research. But we do have with us Dr. Bosic to present her work. Natasha, the floor is yours.
Thank you very much. Can you hear me?
Very well.
Okay.
You can share screen your presentation.
I can share screen.
Okay.
Do you see it?
Thank you. Thank you very much.
Thank you for your kind introduction and the invitation to participate at this interesting conference, because I learned a lot today. And also it's nice to meet you all. And I have to congratulate VALUEMAG participant for successful realization of the project. So I would like to, sorry. I don't know why. Sorry, just a second. Sorry. Okay. So I would like to raise some questions considering first generation bioethanol and to enlighten some possibility to further exploration of starch granule for humans, both in food and non-food application, by using well-known enzyme amylase, but with less known features such as raw starch degrading ability and possible large-scale application, as well as modulation of activity. So in comparison to the fossil fuels, production of bioethanol based on biomass are more sustainable and widely distributed. And currently, there are 3 generations of bioethanol that have been based on different feedstock. First generation is derived from fermentation of glucose contained in starch and sugar crops. And USA and Brazil are the main producers of bioethanol worldwide, utilizing corn and sugar cane, while potato, wheat, and sugar beet are the common feedstock for bioethanol in Europe. However, main drawback of first generation bioethanol is the threat of limitation in food supply, which may affect the human world population as the feedstock are derived from food sources. So second generation bioethanol emerged as a boon to overcome the food versus fuel feud posed by the first generation bioethanol. But second generation bioethanol is produced mainly using lignocellulosic feedstock and agricultural forest residues, and the industrial scale up of second generation bioethanol experienced main hurdle due to some technological issues. Finally, third generation bioethanol is focused on the use of marine organisms such as algae, and algae represents a promising alternative feedstock due to its high lipid and carbohydrate content. easy cultivation in a wide variety of water environment, relatively low land usage, and high carbon dioxide absorption. And above all, algae have a low level of lignin and hemicellulose, which makes it significant to be used in bioethanol production. But even though the research on the application of algal feedstock in bioethanol is still in its, let's say, naive stage, it holds immense potential as a promising feedstock for commercial bioethanol production in future. I have some, I don't know why I have some troubles with the presentation, sorry. So, considering the limitation of first generation bioethanol, European Commission, more precisely Europe's Renewable Energy Directive, RED II proposal from November 2016 suggests a strong reduction of first generation fuels and the replacement by second generation. But those measures are supposed to ensure that Europe fulfills its ambitious climate targets while not endangering food security. Against the background of the ongoing deliberations, Nova Institute conducted a study to compare the sustainability of bioethanol made from different feedstock, most importantly comparing first and second generation fuels. And a comprehensive sustainability assessment shows that first generation bioethanol is as advantageous as second generation for a feasible climate strategy. The results clearly indicate that the systematic discrimination against first generation biofuels of the current Commission proposal is in no way founded on scientific evidence. So criteria selection was based on the most current standards and certification systems of bio-based fuels and materials, including environmental, social, and economic aspects, and a dedicated focus was put on food security. So after analyzing the existing data, performance of the respective fuel options was assessed relative to the others to establish a ranking of the options, and it's based on a traffic light system, as you can see in this table. And this was the analysis of 12 different sustainability criteria, which shows that all of the researched bioethanol feedstock offer significant advantages as well as disadvantages in terms of sustainability. And it comes also for the reduction of greenhouse gas emission, but also when it comes to the negative impact on food security of first generation, the evidence pointed into a different direction because the competition for arable land was counterbalanced by the excellent land efficiency of first generation crops, especially sugar beet and protein-rich co-products, especially for wheat and corn. So in the case of wheat, most of European ethanol production is based on grain of non-food quality and on harvest surpluses, not posing any competition at all, but offering additional outlets to farmers. So in conclusion, we should combine first and second generation to obtain the highest bioethanol yield per hectare, such as first generation wheat plus second generation wheat straw. So the advantage of first generation sugar and starch crops is that they carry the potential of second generation in that by providing their own lignocellulosic co-products without occupying additional areas and at the same time provide protein-rich feed. So I really don't understand, okay, why I have these troubles. Starch is the commonest storage carbohydrate in plants, and it is used by the plants themselves, by microbes, and by higher organisms. So there is a great diversity of enzymes able to catalyze its hydrolysis. Starch from all plant sources occurs in the form of granules, which differ markedly in size and physical characteristics from species to species. Native starch granule is a complex granule composed of two major components, amylose, that is mostly linear polysaccharide, consisting of alpha-1,4 linked glucopyranose residues, and amylopectin, that is more bulky, highly branched polymer consisting of alpha-1,4 linked glucan chains and exhibit about 5% of alpha-1,6 linked branches. So despite large efforts to establish universal model of starch structure, to date, there is no such agreement due to heterogeneous nature and strong dependence from the botanical origin. However, one model seems to be predominant currently, and it is a multi-scale structure consisting of the granule, into which grow rings composed of blocklets made of amorphous and crystalline lamellae containing amylopectin and amylose chains are found. So there are 3 stages in the conventional enzymatic conversion of starch. First is gelatinization, which is heating starch with water to dissolve starch granules and form viscous suspension. Then liquefaction, which involves partial hydrolysis of the starch with enzymes such as thermostable amylase. And finally, saccharification by enzyme involving production of glucose or maltose by further hydrolysis. So for conventional liquid phase enzymatic hydrolysis process, a high energy input is required and it results in increased production cost of starch-based products. But an alternative, known as cold hydrolysis approach, represents a step toward green method for the production of fuel ethanol. And regarding energy cost, effective utilization of natural resources, minimization of the formation of pollutants and viscosity problems, use of raw starch digesting enzymes that can perform direct hydrolysis of raw starch granule below gelatinization temperature is desirable, and the removal of the cooking stage could also increase the value of the co-product since valuable proteins would undergo less thermal stress. The majority of known raw starch digesting enzymes belong to the alpha amylase family, which catalyzes the hydrolysis of alpha-1,4 glucosidic bonds in starch, glycogen, and related polysaccharides. And in our laboratory, we produced and characterized raw starch digesting amylase from one strain of Bacillus paralicheniformis, and that enzyme was capable for direct hydrolysis of cereal, tuber, and root raw starches below the gelatinization temperature of starch and with same efficiency in short duration of time. So having in mind that corn, wheat, and potato are the most important sources of starch in Europe, this enzyme hence is economically attractive. Traditionally, source of starch is potato, wheat, and corn, but also rice, cassava, or other plants which are geographically specific. But also algal plantations are also emerging sources of starchy polysaccharides in past few decades. And algal polysaccharides offer different ratios of branching as compared to amylopectin, which results in not only different texture and physical properties, but also pose new challenges for enzymatic hydrolysis. So some of the issues that had to be addressed before raw starch digesting amylase could become an enzyme of choice for industrial applications were low yield and high cost. But these problems were significantly improved in past years by the efficient upstream processing, smart design of growth media, and optimized production steps, selection of suitable fermentation technique, and efficient recovery processes. Production of proteins at large scale remains one of the challenges in industrial enzymology. Overexpressing enzyme in a widely established host in extracellular form is advantageous approach, and the most important advantage is simpler downstream processing, also increased biological activity and stability, and more quantitative folding and processing. So efficient extracellular expression in E. coli is nowadays possible due to different strategies with various signal peptide sequences. And we reported a new approach based on the fusion of recombinant precursor to the signal peptide of DSBA, which is a well-characterized E. coli periplasmic protein. The potential of this approach was demonstrated by applying two-stage feeding strategy in 2-liter fermenter to achieve high cell density cultivation of E. coli BL21 cells and extracellular overproduction of amylase by fed batch. So this workflow results in higher amounts of target enzyme production, and in most cases can be much efficient than batch fermentation. So designing fermentation conditions in such manner that carbon source exponential stage of growth is constantly added, while care is taken that oxygenation is kept at high level, enables reaching high cell densities. And when the dry cell weight reached intermediate cell density, post-induction phase began, and the glucose feeding rate was kept constant, so enabling cell growth to continue after the IPTG was added. This way, we obtained 0.7 gram per liter of enzyme. And by simple change of expression host to E. coli C43, total amylase reached 1.2 grams per liter. By using mixed mode Novia C prime resin with the idea to exploit hydrophobic patches on enzyme surface, we were able to simultaneously concentrate, remove pigment, and purify enzyme with yield of 96% directly from the fermentation broth in a single step. This method may be useful in the purification of recombinant proteins without tags, since there are many cases where the N or C terminus of a protein is not exposed to solvent, and thus the addition of tags is unfeasible. Also, purification of recombinant proteins without tags is often required for crystallography studies, which was also the case with our enzyme. Since the starch processing industry usually uses the meshes containing approximately 25 to 33% of starch, there is an ongoing interest to explore the amylases for efficient hydrolysis of highly concentrated raw starch suspensions. And there are not many alpha amylases that are able to perform effectively on high concentration of raw starch due to substrate or product inhibition, and because of the need for higher enzyme concentration in raw starch digestion, dictated by mass transfer limitations of the heterogeneous enzyme and the solid starch system. So we have applied response surface methodology as an economical way of obtaining the maximum amount of information in a short period of time for optimization of the hydrolysis of highly concentrated raw corn starch. And the parameters optimized by response surface methodology were the enzyme loadings, the raw starch content, and the incubation time. And what was shown that this enzyme was very potent for hydrolysis of 30% raw corn starch suspension, reaching the final hydrolysis degree of 91% after 24 hours, while hydrolysis can be modulated by changing enzyme doses versus incubation time. Furthermore, it was shown that this enzyme hydrolyzed amorphous and crystalline areas of the granules at the same time, at the same rate, and this is in agreement with a granular by granular mode of attack and can be concluded from the same micrographs with an attacked granule being completely hydrolyzed. The sequence-based classification of carbohydrate active enzymes, CASI database, is the most exhaustive resource of information of glucohydrolases and auxiliary activity enzymes. And CASI classifies alpha amylase in the main alpha amylase family GH13. And generally, enzymes active towards polysaccharides develop different strategies for efficient cleavage, most common being additional binding regions apart from active site to assist the cleavage. So non-catalytic carbohydrate binding is achieved through two different evolutionary strategies. One is additional carbohydrate binding module, which is called starch binding domain in case of amylases, and is often located at separate domains attached to the enzyme by a polypeptide linker, or via one or more surface regions or spots situated on the catalytic domain and turned surface binding sites. Starch binding domain structures and roles have been extensively studied, but there are a lack of information on surface binding sites since those are much less frequently occurring and cannot be predicted from the sequence similarity studies. They do not occur as independent domains and are usually found only by observation in crystal structure of enzyme co-crystallized or soaked with ligands. So the crystal structure of alpha amylase from Bacillus paralicheniformis was determined using X-ray crystallography studies, and it was shown that this tertiary structure is comprised of the 3 distinct domains typical for GH13 family members, also with the catalytic machinery typical for that family, and the active site is located at the center of the catalytic domain and contains a triad of catalytic residues. So the ability of enzyme to hydrolyze raw starch was investigating in terms of the existence of surface binding site, because there is no separate carbohydrate or starch binding domain and structural features that contribute to this high activity with raw starch, and because it was unclear. So we seek to elucidate whether the highly efficient raw starch digestion activity was influenced by other interaction with starch, by presence of some spots on the enzyme. So using X-ray crystallography studies with different ligands, we identified a surface binding site on the surface of the catalytic domain of enzyme at the N-terminal end, and with 2 key amino acids, phenylalanine at position 257 and tyrosine at 358, which was involved in forming the complexes. So starch surface binding site was located on a groove on the surface of the enzyme, which role is most likely localization of the enzyme on the substrate, enabling the active site to perform multiple catalyses without releasing enzyme from the enzyme substrate complexes. So the role of these surface binding sites in starch binding and degradation was investigated by kinetic studies with different substrates and with enzyme variants carrying mutations in the surface binding site region. So the 2 most important amino acids were mutated both individually and simultaneously to obtain variants of enzyme, in which case the key amino acids were replaced with alanine. So this approach was aimed to remove hydrophobic stacking interactions with carbohydrates observed in the crystal structure with oligosaccharides. And mutagenesis is an effective tool for eliminating binding at these sites, and then which allows evaluation of the impact on activity and binding characteristics. Kinetic studies with different substrates, including raw starch granules, show that starch binding through this surface binding site is disrupted when with each amino acid contributing cumulatively to binding and hydrolysis. Mutation of both sites resulted in 5-fold lower efficacy for raw starch as a substrate and 5-fold weaker binding compared to the wild type enzyme, and suggesting that the ability of enzyme to hydrolyze raw starch with high efficiency is related to the level of its absorption onto the starch granule. This enzyme can be further exploited for its robustness by altering its activity and converting this hydrolase into transglucosylase. The use of transglucosylases for synthetic purposes has been limited since these enzymes are relatively rare in nature, and the ones that have been characterized act on a limited substrate repertoire. By contrast, hydrolases are extremely common and act on a wide range of substrates. So to alter the activity of amylase, histidine at the position of 235 was replaced with glutamic acid, And the mutant enzyme was characterized in terms of its mode of action using different substrates such as starch, amylopectin, malt oligosaccharides. And the mutant exhibited high transglucosylation activity, while the wild type exhibited only hydrolytic activity. So converting hyperthermostable amylase into transglucosylase yields a highly potent tool in synthesis of the starch derivatives. Yeah, finally, I would like to thank all of my collaborators, which I was realizing this project through the years, and from my university and the university from abroad. And also, I would like to thank you for your attention.
Thank you very much, Natasha, for your interesting presentation.
Yes, thank you. Thank you, Natasha. This is Angelo.
Okay. Thank you, Angelo.
And I hope to see you soon in Greece, also in Athens, to arrange possible collaboration in this field of.
Yes, because the enzyme seems very promising, and both also on the microalgae. Because I don't know if you remember. I think we can start now discussion.
Yes, we can.
The presentation of Patrizia about the exhausted microalgae biomass after astaxanthin extraction. The exhaust biomass contains a lot of sugar, let's say, and other fibers that are difficult to be, let's say, used straight. Maybe the action of this enzyme can release much more, let's say.
Yeah, this is one of the example of glucohydrolases that we are working on. We have a lot of different, not only active to starch, but also active to cellulose or the hemicellulose and lignin even. So, but okay, algae don't have this, but it's okay. But for starch also we have a great diversity of enzyme acting on different parts of starch. So probably something, but even maybe this transglycosylase activity is also very interesting, because maybe we would be able to obtain some possibly new dietary fibers, because this is like a topic now to have some polysaccharide and try to make a different one from this one by using transglucosylase. So I think the possibilities are large.
Okay, this is good. This is good for us to enlarge first the research field, but also to propose new, because PBI in particular, but also the Horizon Europe is going to ask for innovative products, new products with less impact on the environment. So I think there is room to collaborate and to go forward.
Yes, I hope so.
Yes.
Okay, thank you, Natasha.
Thank you very much.
The point is that we have questions from the audience concerning the VALUEMAG project, and let me start with the easy things. Easy things concerning, for example, the question on disseminating the presentations. I think that from the point of view of VALUEMAG participants, VALUEMAG partners, I think that the presentations are, let's say, available. And I kindly ask from Annabelle, from the moderators, to send to the audience the presentations, if it is possible.
Yes, thank you for your comment. Indeed, all the presentation will be sent to the participant. We just would need to receive a version that we can disseminate to all participants from the speakers. Then in the next few days, please, if all the speakers can send us their presentation, making sure that all the information in it is, let's say, diffusible, and then we will share, let's say, the presentation with all the participants.
Okay. Okay, fine. This is excellent, Johan. Thank you very much. Now, concerning the questions, there are 13 questions asked by audience, and they have been answered by written answer. I don't know if someone wants to come back to them. And there is also another one, an open question from Nicola. I observed the discussion between Nicola and Dr. Molino and Antonio. I don't know if you want to say a few words on these questions. Also, there is a question concerning, and not answered question concerning the last question, the 14th question, considering important volume of molecule of interest for feed above 1 to 10 kiloton per year, how industrialization scalability, I mean, and capex investment.
Okay, Prof.
Yeah.
I'm ready. Okay, the question is very simple. In particular, in the framework about the task 3.8, we have done an evaluation about the price of carotenoids production, but and the value obtained was about of 400 euro with VALUEMAG tech for each kilogram of carotenoids production. But sure that this value taking in consideration only the process cost and not the plant cost. I suppose that the selling cost could be greater than this, and on this direction is necessary to do a feasibility analysis about the integrated VALUEMAG plant. So about the question, in this direction, the choice of the sites for integrated plant, considering the watering, drying, and extraction is very important in order to minimize the cost for the sale, and thus making them competitive with the current cost for these biomolecules.
Correct. I think that such a feasibility study must be one of the objectives of the forthcoming projects following the VALUEMAG project in order to industrialize actually out of this.
Yes. But in this direction, we are on the right way because only considering the process costs that are the higher cost, the price for beta-carotene is lower than the market prices. And therefore, I suppose that the way is right.
Okay. Now, concerning the 13 questions answered by presenters, mainly by Angelo and Antonio, or Antonio and Angelo, I think that from my point of view, there are some. There is another question concerning Fenton reaction. Does anybody want to answer on it?
I don't know really what is the Fenton reaction. That's why I didn't answer.
Well, actually, I am a magnetician, so far away from my topic. Well, I'm afraid that this answer, I cannot answer. If anybody else from the panelists can answer, she is very welcome to do so. Otherwise, I'm waiting for other answers from the audience, and particularly if there is an answer that is not satisfying, then please repeat question and in order to proceed.
Vangelis, I think there is one question that we didn't answer from
Angelo, please.
Yes, from Florent. I think. About the beta-carotene, okay, Antonio answered, yes, but I want just to, if it's more convenient to extract beta-carotene, lutein, algae, and other algae extracts from microalgae compared to plants. Antonio explained it, but I want also to add something else. I don't know if he's still in the audience, but when we talk about microalgae cells, we talk about something that is already clean. So compared to other crops that grow on land, we don't have roots, we don't have leaves, we don't have branches. So all the biomass is concentrated in these very small cells, but full of stuff that can be used almost 100%, as we demonstrated. So compared to crop, compared to other plants that lives on land, microalgae are superior because, as I said, they don't have almost waste. Everything can be used in an easy way. So this is just an integration.
I also want to comment on one comment of Nicola from Extratex. It could be useful to discuss the possibility of experimenting, let's say, on this supercritical CO2 equipment, which could actually be a possibility for collaboration, actually. From my point of view, I don't see, actually we didn't answer the CapEx investment for such a production mentioned by Nicola, but I think this is a matter of the feasibility study that Antonio mentioned before, and we can do in case we are interested on such a estimation. Does anybody else from the panelists want to give an answer to the questions raised by the audience, please?
Okay, again me.
Yeah, yes, Angelo.
Yes, because there was also a question about the daughter cells, the cells that are generated from the growth, if they have or not magnetic nanoparticles. I answered, but I think I didn't stress this on the presentation. Actually, what we get with this magnetic immobilization is that there will be always a parental population that is stuck on the cone and produce, let's say, new cells. And the new cells, they are washed away with the flow of water that constantly runs on the parental cells. And once a day or once every maybe 30 hours, these cells are removed from the cone. So, and new nutrients are injected. In this way, the parental population is always in the best condition to grow and to generate day by day new cells. I want to add just the last thing. This is very important for Hematococcus pluvialis, for example, growth, because Hematococcus, as you have seen in the presentation, produce astaxanthin, but it doesn't produce astaxanthin immediately. It needs to grow through a sort of stressing phase that can last 1 week or 10 days, where the amount of astaxanthin goes from 2-3% up to 20% of the weight of the cells. So doing this in a normal photobioreactor will compromise, let's say, the growth, because the starvation will affect everything. In our case, there will be a population that generates cells, and then these cells are removed and then can be starved, and astaxanthin can be produced continuously, let's say.
Okay, good. Any other panelists to add something? If not, then I kindly ask the audience to see if there is any more questions on the presentations. At this moment, I don't see any other question. Johan, Annabelle, I would like to put the floor to you to see how we proceed now.
Okay, thank you, Evangelo. Congratulations for this very interesting project and that arrive at the end. I hope it's not the end of the technology and it's just the start, let's say, of the industrialization, because I've been following the webinar during these 3 hours, and indeed it deserves to have a follow-up and maybe to raise a new BBI or CBE project in the coming calls. And should you like to have, let's say, the help and the insight of our cluster IR, feel free to contact us, and we will be very pleased to help you to continue. Industrialization is something very important. It's something that we follow, and we have been very successful so far. I would like to thank as well VALUEMAG, you, Evangelo, and specifically Nick for, let's say, contacting us to organize your workshop, because at the beginning it was supposed to be a workshop. Unfortunately, the sanitary conditions have decided otherwise, and we transform it, and we thank you as well for your flexibility to transform it into a webinar. But I think it was the best thing to do, because we are not rid yet of this virus, and for a project like yours, if you want to go ahead and to comply with your objective in communication, workshop, and so on toward the EU Commission, it was very necessary to transform into a digital session. Thank you, Nick, for that, and I hope you are satisfied with, let's say, this webinar, and hope you get a lot of insight and news and comment and possibilities to follow up this project, and a lot of interest from the audience. Nick? EU
yes.
Commission.
Thanks very much, Johan. It was a very good organization of this webinar.
Thank you, Nick,
of course, VALUEMAG next year will not be a project, an active project. I hope to have the opportunity to visit the next BIOKET as a new consortium. Or
we are still probably having YouTube open. From the audience.
Excuse me?
We are still hearing my presentation or something behind you when you're speaking.
Okay. Just
here with Vangelis. Okay, so thank you very much for all of this support and the organization of the webinar. Next year, of course, VALUEMAG will not be an active project, but I hope to be the continuation of this project and to have the next step for the VALUEMAG and to visit BIOKET. I would like also to thanks BBIGU for their support and the funding of the project, and all the person behind the scenes. Our project officer is in the audience of this webinar, and thanks a lot for the help and the effective participation and help to all the issues. And I hope all the audience to be satisfied with this webinar, and we are open, and we are looking forward to receive more questions or further opportunities for collaboration and exploitation of our project results.
If I also, if you excuse me, if I, Johan, your microphone, please.
Yeah. I know as well that some of you will be participating to the B2B this afternoon. Then again, I would like to remind to the participants, those who are registered to the B2B, that they will have opportunity to meet a few participants of VALUEMAG during the B2B, and it's another opportunity to ask questions. Sometimes it's easier to make it on face-to-face than via a webinar and so on. And maybe there will be some new opportunities of cooperation, new ideas for, I don't think it will be the same name, but we can call VALUEMAG 2.0.
If I may add a few things, I think that we have developed three main technologies. The first is the magnetic cultivation of microalgae. The second is the selective magnetic separation. And the third is the targeted, the magnetic algae means that they are alive when we put particles inside. So it may result in three products. Product number one is cosmetics with Astaxanthin. If we achieve the rejuvenation creams, just excellent. Second is the pharmaceutical applications like magnetically targeted drug delivery, which is a very hot topic, and of course also food out of the residues or the algae itself. So we are, thank you very much for the hospitality. Nick expressed already the gratitude for the BBI and your hospitality, and hope to see you soon under a new scheme, for example, a new demo or a new coming project it
is the baddest thing I can propose you for the future, indeed, to be ready for a demo.
Thank you very much. Thank you very much.
And especially to be a real event and not through the web.
Through the web.
Yeah, exactly.
Other from close. Yes.
Thank you very much.
Thank you. Thank you very much indeed,
guys, for this kind of conclusion. If you don't have anything else to say, I just want to address a few words to the audience. Thank you for having participated to this first webinar of BIOKET Digital. Again, we did our utmost to be as much flexible and agile as possible under current situation. Tomorrow we have a second webinar in a total different sector. It will be on phenolic acids, a very interesting sector anyhow for bioeconomy. And we hope you will be a lot to participate. On paper, we have more than 250 participants tomorrow. Again, don't forget to look at your email box, because you might have a last-minute request for a meeting this afternoon during the B2B. We have 15 hours of B2Bs. 5 hours this afternoon, 5 hours tomorrow afternoon, and 5 hours Friday afternoon. Still time to register if you want, still time to make appointments. Make sure you follow up your mailbox to reply to the request you would receive. And we do hope that there will be a lot of business opportunity, partnership opportunity. And I would like to thank again VALUEMAG and all the speakers of VALUEMAG, and maybe those who are still behind their camera, they can open up the camera to just the last word. because we can, maybe someone wants to do a print screen to see it's like a photo, family photo for this webinar. Then everyone can open the camera. And I would like maybe Evangelo, one of you, for your records, to make a family photo of a VALUEMAG workshop. It's not the same than
the normal one,
but at least it has the merit to exist.
Indeed.
Okay.
Okay, guys, thank you very much. And I hope to see you next year in Lille for BIOKET on site. And hopefully we can meet each other somewhere in our life for different projects or VALUEMAG 2.0. And thank you very much again. And see you tomorrow. Thank you.
Excuse me.
Yes.
Oh, okay, nothing. It was a question, but actually it was a thank you message from a colleague.
Okay, perfect. Thank you very much.
Thank you.
Nice. And
Thank you.
it's time for lunch break before the B2B.
Yes, yes, of course.
Bye.
So, bye-bye, everyone.